A student asked me a question: how do I use my book of distinction material to get a distinction? That I said that whatever question is being asked is being indicated in that book, and asked me that I should use 2022 National Senior Certificate Examination to answer that question. So let me show you exactly as we are comparing the question versus where do I find the answer in the booklet. For this case, I will only focus on questions not related to objective. So on the side of the objective, I'm going to be a bit faster. Let's start with the section A. The component of DNA molecule that provide the code for protein synthesis. Which component of DNA molecule that provide the code for protein synthesis? So what causes this are the nitrogenous bases. What are the nitrogenous bases? We are talking about the thymine, the guanine, the cytosine, and the adenine. These are the nitrogenous bases which are supposed to be organized together to form a gene. So these are the ones which bring about a protein. So the answer is D. Then 1.1.2, during which stage of meiosis do spindle fiber begin to form? So the spindle fiber always begin to form under the preparation phase, which we call the prophase. So it means that the answer is going to be A. I'll explain more when we go to other sections. Then they're saying that an individual that has Down syndrome in the karyotype, there is an abnormal number of chromosome at chromosome pair. So Down syndrome, whenever you talk about Down syndrome, yes, so your mind must go to chromosome number 21. Whenever you talk about Down syndrome, so you must think about an extra chromosome on chromosome number or chromosome pair number 21. So the answer is going to be C. So don't forget that these objectives, each objective is 2 marks. Then they're saying that an individual that receives an identical allele from each parent is described as, how do you describe an individual that receives an allele which is identical from both parents? For example, you have the parent is capital T, small t, and then another parent is capital T, small t. So now, if I receive this and this, how do I call this? So now, if the baby gets small t, or this and this, and the kid is capital T, for example, yes, it means that these 2 alleles are the same. These 2 alleles are the same. So how do we call that individual which receives those 2 alleles? So because they are the same is homo. So it is going to become homozygous, because you are describing a zygote, yes. So that zygote has the same kind of allele. So the answer is going to be C, meaning that homozygous. Then the next question, they are saying that the plant species that has a diploid chromosome number of 12. So if a plant species has a diploid chromosome number of 12, yes, which of the following is a haploid chromosome number for this species? It means that if it's diploid, di means 2. Di means, if it is diploid, it means 2. That's why it's called it 2 sets of chromosome, 2n. So haploid, diploid means 2, and then haploid means 1. If the diploid is 12 and this means 2 sets of chromosome. Then haploid means half. Yes, so half of the 12 is 6. So 1n is going to be what? Is going to be 6. So when we come back here, we shall see that the number of chromosome from 12 is going to be, half of 12 is going to be 6. Yes, so it's going to be 6. So the answer is going to be C. Then they are saying that inheritance by multiple alleles. Inheritance by multiple alleles in genetics is referred to as 2 alleles that influence the 2 characteristics. 2 alleles, no. It's more than 2. More than 2 alleles, this is correct, that influence 1 character. Yes, it is more than 2 alleles. Then those 2 alleles must influence those 2 alleles must influence 1 characteristic. A good example for this is blood group. Yeah, it is 1 allele, it is 1 gene. Which gene is blood group? That's 1 character. And then how many alleles are there? We have allele for A, dominant allele for B, dominant allele, and then allele for O, recessive allele. So we have how many alleles? There are 3. So because there are 3, there are multiple. These multiple alleles will influence 1 gene or 1 characteristic, which is blood group. So that's why you're saying that B is the most correct answer. More than 2 alleles that influence 1 characteristic. Which one of the following is a reproductive isolation? When you talk about reproductive isolation, it means that the organisms are being isolated as a result of the reproductive strategy or the reproductive mechanism. So they're saying that breeding at the same time of the year. They can't be isolated if they are the same time of the year. Infertile offspring. This one is correct. Plant adaptation to the same. If it is the same pollinating agent, then they can't be isolated. Improved fertilization. This just increase the ability to bring about fertilization. So the answer is going to be B. Why? An example is, for example, a horse. You have a horse and then a donkey. When you cross the 2, you've produced a male which cannot reproduce again. It is infertile. So it means that they are being separated. So the answer is B. Then you're saying that normal human ova have normal human. Remember that when you talk about humans, they have what you call 46 chromosomes. Yes, but each ova or sperm, it has half of the chromosome number. Yes, so it means that it has 23 here, 23. But among these 23, if it is a male, it's going to have Y, and if it is a female, it's going to have X. But which chromosome number will have the Y and which chromosome number is going to have the X? The chromosome number 20, the 23rd chromosome is going to be X on the female, and the 23rd chromosome is going to be Y. 23rd is going to be Y on the male. So that if now the Y of the male fuses with the ovum of the female, which is X, is going to be XY, and then we produce a male. And then the female is always X, so that's why the X of the male fuses the X with the X of the female, then it's going to become XX, and then it's going to become a female. So now they're saying that normal human ova have 22 autosomes. From chromosome number 1 until chromosome number 22, we call them autosomes in both male and female. So 22 autosomes. So whenever I have 22, that answer could be correct. I have a 22 here, I have a 22 here. So it means that now these two answers are wrong. So we have wiped them out. So it means that I have this and this. But they're saying a normal human ova. When they talk about ova, they are talking about a female. So this must go to female. Yes, if it is a female, female can't have Y. They're always having X. So it means that the answer is going to be X. And 22 autosomes and an X. It can't be 22 autosomes. Yes, it's 22 autosomes and a Y. Then it is going to become a male. It's going to become a sperm. So it is 22 autosomes. So basically it is an ova. It is not a sperm. Then the next question is saying that which one of the following occurs in mitosis, but not in which one of the following occurs in mitosis, but not in meiosis? When you talk about mitosis, mitosis, you have one cell. You have one cell. One cell will divide to form two cells, but these two cells are identical. But if you look at meiosis, you have one cell, you form two cells in the first meiotic division, you divide again, you form four cells. These are the one which forms the sperms and the ova. And then these have the chromosome number. They have half the chromosome number to the parent, which is, for example, if it is a human, is 46, and then here will experience 23 chromosomes. So now what happens here in the question? They're saying that two cells are formed at the end of the division. They're saying which one of the following occurs in mitosis. So it means that it occurs, we are comparing mitosis over meiosis. It means that the first statement must be belonging to mitosis. The second statement must be belonging to meiosis. So the first statement, they're saying that mitosis, they're saying two cells are formed under mitosis at the end of the division. Yes, we see here two cells are forming at the end of the division. They're saying crossing over takes place. We don't have crossing over in mitosis. We don't have crossing over. So this one is ruled out. The most correct answer is A. So the answer is going to be A. And then the last objective is about the graph. Is about the phylogenetic tree. They're saying that the graph below shows the percentage similarities between humans, so we are not talking about differences. We are talking about similarities, percentage similarities between humans, and then DNA, and then DNA of some species of African apes. So it means that if you look at this, if it is similarities, it means that humans and the chimpanzee will share 98% of DNA. And then gibbon and the humans, they share 96% DNA. And then the rhesus monkey, we share 94% DNA. And then the galago, we share 72% DNA. So now a question is asking, which one of the following phylogenetic tree best represents information in the graph? They are saying that basically, if you look at these percentages, the more is shooting towards 100, the more is like we are related. It means that humans and the chimpanzee, we are more closely related than humans and the gibbon. Why? Because the difference here is 2, while the difference here is 4. And then the difference here is 6. It means that we are more closely related. If we look at diversion, we diverted recently compared to the gibbon. Now let's go to, we are seeing here human and chimpanzee. Yes, they are a little bit closely related and they are recent. If you look at them here, 2%, difference and 4% difference. And then it means that we are recently, but we are next. The gibbon, chimpanzee, yes. The gibbon diverted after. No. Before the chimpanzee diverted from human. This is what it means, that the gibbon diverted after. No. Before the chimpanzee did what? Separated. And then the rhesus monkey, because it is 6%, was the first before the gibbon separated. And then if you look at the galago, separated a long time ago. That's why you see that the difference between this and the human is too big. Why? Because they diverted long time ago, so the nature modified them differently. So if look at the most correct answer will be C. You'll find out that the most correct answer is C. This is a phylogenetic tree which we are requires to be studied a little bit more. And if you look at it, you don't need to cram, you just have to know the principles of understanding of the principles of the phylogenetic tree. How do you apply this phylogenetic tree? Basically, you just have to look at the similarities and differences. Then these are 10 questions. In most cases, they come between 8 to 10, and each question comes 2 marks. So now we're going to go to question 1.2, and this is related to the terminologies. 1.2, which are biological terms. Here is saying that the division of the cytoplasm. Now here we are going to see the answer corresponding where we are supposed to get these answers from our booklet. As we said that it is a distinction material, so we need to find out each and every question which is in this paper. Where are we going to find the answer? This division of the cytoplasm. This is called cytokinesis. So where do we find this? How do we know this? So we're gonna go to our book, and then you go to meiosis. Yeah, it's here, telophase. We are saying that the daughter cell chromosomes reach the pole, a new nucleus form, the cell membrane of each cell constrict. The cytoplasm divide into cells, which we call cytokinesis. So meaning that division of the cytoplasm is called cytokinesis. Here, that's how it is being answered. And then we go to the next question, the sugar molecule present in the nucleotide of RNA acid. It means that the kind of sugar which is found there is ribose. So where do we find this? We go back to our book on DNA, we're gonna find it here. You're saying that single-stranded molecule consists of nucleotides. Each nucleotide is made up of a sugar called ribose. So you see the answer is there. And this is the structure of RNA. So ribose is the sugar which is located in RNA. The position of a chromosome, the position of the gene on a chromosome, this one is called locus. So if you go to our book, where do we find the position of a chromosome? We'll find it at the beginning of genetics, position of the gene on the chromosome. So basically, these are what we call the locus. And also here, you see that these are positions of genes. So each individual position is called a locus. And if there are many, we call them loci. Another question, they're saying that the process by which DNA makes exact copy of itself. This is DNA. This is what? DNA. We'll find out that it's called DNA replication. I think this one is easy. You can find it in DNA replication. What is DNA replication? Is the process by which DNA makes as an exact copy of itself. And if you look at this question, they are saying the process by which DNA makes exact copy of itself. So exactly what is there in that book is just what they brought here, and then they're asking you the question. So it is DNA replication. undifferentiated cell that may be stimulated to develop into any body cell. So we call these ones stem cells. So where do we find this? We'll find this in our book in genetic engineering. Yeah, there we go. You're saying that stem cells, these are cells with unique ability to develop into specialized cell type in the body. So if you look at the question here, it's saying that undifferentiated cells that may be stimulated to develop into body, into type of body cell. So here you're seeing that these cells, what happens? They can develop into specialized cell types in the body. So it's just a reverse, but that's what they are looking for, which you call the stem cells. So there is no question which you're going to find in the booklet, you're going to find in the exam, and then it's not being answered by this booklet, which we call a distinction material. Excel in life science. So now there is another question. They're saying that Mendel's principle, which states that the organisms possesses two factors which separate that each gamete contain only one of these factors. Why do we call them factors? Before we started to call them genes, we are calling them factors. The answer here is Mendel's law of segregation. So basically, that is the answer. It's a segregation. So now, where do we find this answer? We will find this answer from this booklet. Yeah, we'll find this answer on this book. Mendel's law of segregation states that they're saying that Mendel's law of segregation states that the diploid organisms passes randomly selected alleles for a trait to its offspring, such that offspring receives one allele from each parent. So the question here is asking, the principle which states that organism possesses two factors which separate. Yes, they separate so that each gamete, that is the offspring, each gamete will contain only one of these factors. So here they are saying that the diploid, it means that the organism passes randomly. Yes, we don't know which one is it, but randomly, it happens randomly. The selected allele for a trait into offspring. How do you pass this? We pass them through gametes, such that the offspring receives one allele from each parent. So this is Mendel's law or principle of segregation. So our answer here comes back to Mendel's principle of segregation. Then another question, they're saying that evolutionary theory that propose long periods where species do not change, alternating with the short periods where rapid change occurs. This is punctuated equilibrium. Where do we find this? Yeah, the answer is here. It's saying that punctuated equilibrium, what happens to it? This evolution involves long periods of time where the species do not change. So it alternates with, it has short periods of time where there is a rapid change. There is evolution, there is long period of time where the species don't change, and it alternates with the short periods of time where the species is changing. So basically, long period of no change and then short periods of species changing. If we come back to our question, we shall see that they're saying that theory that proposes that long periods of species not changing and alternating with the periods where the species is changing and it is short period. So basically, that is the answer. So this is also being answered by the book. Tangled network of DNA protein located in the nucleus. This is the chromatin network. Let's see where we find it. Yeah, we can find it here. Chromatin or chromatin network is being indicated here. Yeah, here is showing you that it's a substance within chromosome consists of DNA protein. So if you look at it, you see it that it is a network. It's a network. And also, if you go back to DNA replication, you'll find it again. It's chromatin network, how it is consists of. You see, these are what you call chromatin network. These are the chromatin network. So basically, still it can be answered by that. So you're saying that entangled network of DNA. So it means that it consists of DNA and protein located in the. So if you look at it, it is DNA, yes, and its network and is located in the nucleus. Then the natural shape of DNA is double helix. How does our book answer this? If you go to the structure of DNA, yeah, you have to go to the structure of DNA. Yes, describe the structure of DNA. You will find out that DNA strands twist to form a double helix. Forms a double helix. So basically, the book answers that question also. Then another one is the phase in cell cycle during which cell growth occurs. During which cell growth occurs. When does cell growth occurs? We shall see that cell growth occurs in interphase. So where do we find this in our book? So in during this is what called the growth phase. And then we have the mitotic phase. So this is being answered by during interphase or before the cell division. So basically, if you look at DNA replication, that's when the cell is growing. It's supposed to grow, and then the growth occurs during interphase or before the cell starts to divide. So this can answer our question. A group of similar organisms that in a particular place at a particular time with ability to interbreed. What is it? A group of organisms that occur in a particular place at a particular time with ability to interbreed. Since it's a group of organisms, then it's gonna be and they're able to interbreed. Since they can interbreed, then automatically it is a species, but it's a group of these organisms. Then because it's a group, then it's going to become a population. So the word group cancels out for the species and then forms population. So the answer becomes only. They are saying that manipulation of biological process to satisfy human needs. The answer is because it is manipulation, it is technology, but because it is biological process, then it's bio. So it becomes B only. Who discovered the structure of DNA molecule? James Watson and Francis Crick. Francis Crick, then it means that both A and B. Please always follow instructions, because the instructions saying A only, B only, both A and B or none. Some people, they only write A, they write B, they write both. What is both? Both mangoes and oranges? Both males and females? What is both? You have to say both A and B. Don't say A, because if you don't just say A only, it means that maybe even B is correct. What you do is you have to put only, both A and B or none. Try to follow the instructions. In tomato plant, tall stems are dominant over short stems, and the red fruits are dominant over yellow fruits. A farmer crosses a homozygous tall, then it means that now it is homozygous tall, so it means that it's going to be capital T with the yellow. Yellow is, we say that yellow is recessive, which is the moment they say yellow, I don't need to say homozygous or heterozygous. It's always recessive because it is always heterozygous. sorry, is always homozygous because it's recessive. Plant that is heterozygous for both characteristics. A farmer crosses homozygous tall, yellow, yellow tomato plant with a plant that is heterozygous. It means that heterozygous for both, then it means that it's going to be capital T, small t, and then capital R, small r. Then name the type of genetic cross. Because you're involving two characteristics. Which characteristic? The height of this means that we are involving the height, and then crossing with the another characteristic is the tomato color. That is the color of tomato. So two characteristics, that is height and the color. So the answer is dihybrid cross. Where do you get it in our booklet? Yes, we get it from here. Dihybrid cross involve inheritance of two characteristics. So yeah, we see that you have height and color, two characteristics. So we're saying that in the dihybrid cross, a cross which involve two character. And then they're saying give the genotype of homozygous tall and yellow plant. We have seen homozygous tall, we have seen it, which is capital T, and then yellow, which is recessive. We said it here is recessive, so it's going to be small. List the genotype of all of possible gametes for a plant with heterozygous both alleles. So if it means that if it is heterozygous, it's going to be capital T, small t, capital R, small r. So that because it is dihybrid, it means that I'm going to have this with this. It's going to be capital T, capital R, this with this. It's going to be capital T, small t. Then this with this, it's going to be small t, capital R. This with this, it's going to be small t, small t. That's why there are 4 marks, giving you this, giving you this, giving you that, and that. Yes. So we have this must go with these 2, and this must go with this 2. So this T with capital R, then capital T with small r, then small t with capital R, then small t, small r, small t, small r. Yes. So basically that is it. Then they're saying that in hemophilia, hemophilia is a sex-linked recessive, is a sex-linked trait. The pedigree diagram below shows the inheritance of the hemophilia in the family. Now, you always need to know that if it is sex-linked, even if they say male and female, but they have not mentioned that it's a sex-linked, please don't use X and Y. But if they say sex-linked, always use for a male, always put XY, while the female use XX. And you also need to know, before you go for this question, you need to know that always, because a circle, a box means a male, while a circle means a female. Sometimes we don't give you a key. Sometimes we can even ask you a box means what? Yes. So let's continue to our pedigree. Can this question be answered by our booklets? Yes, because we have many questions concerning about pedigree diagram. Let's see. Yeah, this is a sex-linked disorder. So it means that whenever you see a sex-linked, a box means a male, while a circle means a female. All right, let's go back to the question. State what is represented by a square or a box on the pedigree diagram. We have seen it that a square is male, while a circle is a female. State the number of generations represented in this diagram. You have to count them. 1, 2, 3. So those are 3 generations. 3 generations. Just count 1, 2, 3. those lines. Then you're saying that offsprings of individual P and Q. P and Q. So these are offsprings. However, not whatever on this line means offspring. This gave rise to this, and this. What about this and this? This one is an intruder. Who married this guy? Married this girl from these people. And also this girl married this guy from these people. So these are just people who came into the family, but these are the offsprings of P and Q. So there are three of them. They are saying that give the letter only of female who have hemophilia. Letters of female who have hemophilia. So the shaded part is because it is recessive, they are affected with hemophilia. So now they are saying that give letters only of female who have hemophilia. So female is 1, is 2. So there are 2 people who have hemophilia. Then give the genotype of R. R. Where is R? R is a female, but a female came from a guy who had hemophilia. So it means that it is heterozygous and is a female. This one and this one, they gave rise to a baby who is sick with hemophilia. Then it means that the lady is heterozygous. So it means that R, since it is sex-linked, because it's sex-linked, they mentioned it. So I'm going to start with X and X because it is a female, and then hemophilia is caused by a recessive allele which is put on the X chromosome. So for normal, it's gonna be capital H because, and then small h. Why? Because this person is heterozygous, is a carrier who carries this recessive. Because we have to see how did they get this. If a male, if this guy is capital X, small capital X, capital H, because it is normal. Y in the moment, Y doesn't carry anything. And then this one X, capital small, then it's capital H, small h, so that now this combine with this combine with this, and then I form capital is X, small h, and then Y, which gives rise to this boy. Therefore, the female, if it was capital H, I wouldn't have produced the baby who is sick. So the genotype of R is capital H, small h. Then another question, this marks the end of section A. What about section B? What about section B? You see that the questions in section A, you can score all the marks. You can score all the marks. Question 2, they are saying that the diagram below shows or represents one cell in a phase of meiosis. They are not saying that this is a human cell. They have not talked about the word human. So you must not think about 23 pairs of chromosome, please. Don't talk about 46 chromosome, please, because they have not talked about human. So now they are saying identify the phase of meiosis. So they are along the equator. Along the equator. So because they're along the equator, so the phase is gonna be metaphase. But which metaphase? They are single. They are not double. If they had another one here, then they would have been double. So it would have been metaphase 1. But because they are single, therefore it is metaphase 2. So it means that the phase which is indicated there is metaphase 2. Give an observable reason for your answer because chromosomes have been aligned along the equator singly. So that it must show. Where do we find this? In our book, we go to meiosis. Yeah, you see that the chromosomes are aligned along the equator singly. But if it was supposed to be meiosis 1, it would have been like this. There are 2, they're in pairs, but they're not in pairs, therefore it is meiosis 2. Then they're saying identify A. What is A? A is a cell membrane. Don't confuse the cell membrane with the nuclear membrane. What is outside is cell membrane. And then B, these are fibers which hold the chromosomes in place, which are spindle fibers or spindle threads. Then describe the role of B in the movement of chromosomes during meiosis. B. These ones, they are very important as they try to contract, they shorten, and then they pull the chromosomes or the chromatids to opposite poles. And also, they are very important in holding the chromosome in place, maybe during metaphase. So where do we get this? We get this during anaphase. Spindle fiber shorten and pull the homologous chromosome to opposite poles. That's where we get that answer from. What? Spindle fibers or threads are the one which hold these chromosomes. So the book is answering that question very well. Then another question that's saying that draw a diagram of draw a diagram of the draw a label diagram of the structure C that would appear in final phase of this mitotic division. Show the correct shading of C. So where is C? So remember, this is gonna split. It is a chromosome. It is a chromosome. So if it splits, let me draw it here. It is a chromosome, yes, with a centromere there, but half of it is shaded here. We see it. So when it splits, one is gonna go this side, one is gonna go that side. So it means that this chromosome is gonna look like that, and then the other one is gonna look like that with a shaded part. So what is this? When it reaches the pole, we call it chromosome. Chromosome. But which chromosome is this? Is unreplicated chromosome or single-stranded chromosome. Or we can call it daughter chromosomes. Yes. Whenever I ask you to draw, never ever draw anything without writing a title. You must always write a title in there or caption. The next question is about DNA. This DNA is basically under protein synthesis. So, can our book answer this question? Let's go there and see. Yeah, the question is there. That's where the question is. The question is here with us. It's there. And then here, there we go. They're saying that. Name the process that occur at W. This question. This is what they're looking for. So if you come here, they're looking for this. And then we also have the a question. We have this. This is the question. They're looking for what is happening inside this process. This process, if you look at this, what is happening inside the nucleus. That's what they are looking for, because this is the nucleus, which is transcription. Then they're saying, name the process. Why? Obviously, if you have transcription, this one is going to be translation. So they are looking for this, translation. Then they are saying that identify organelle X. Organelle X, this. They are looking for this organelle X, this wall. This wall. If you come here, they are looking for this wall, which is the nuclear membrane. Remember? Remember, this is the nuclear membrane, the membrane of the nucleus, while this one is cell membrane. Yes. And then they're saying that molecule Z. Where is molecule Z? Molecule Z. They are looking for this. Because it is on the ribosome and it has moved out of the nucleus, is this. Then it is messenger messenger RNA. Yes, and then they are saying that state two locations of DNA in a cell other than in the nucleus. They're saying two, we find it in the mitochondria and also in the chloroplast. So let's see it in our book. If we come here on the types of DNA, we shall see that if you have the nuclear DNA found in the nucleus, they say other than this. So we have the mitochondrial DNA found in the mitochondria, and then the chloroplastic DNA found in the chloroplast, meaning that that is the answer. Then another question they're asking, describe the process W. What is process W? They're looking for transcription. How do I explain, or how do we explain transcription from our book? Transcription is being explained nicely. DNA double helix unwind. So basically, we give you a tick here. DNA double helix. Double helix, in most cases, we give you a tick. Yeah, DNA double helix unwind. So when you say unwind, we give you a tick here. The weak hydrogen bond break. Weak hydrogen bond breaking and unzipping is the same thing. One strand. This is very important. Sometimes they say two strand. No. One strand will act as a template of messenger RNA using the free messenger RNA nucleotide. Then they're saying that the messenger RNA is complementary. We like this, complementary to DNA, whereby A goes with U and then C goes with G. Remember, this is transcription. You are forming messenger RNA. So we don't have messenger RNA now has the coded message for protein synthesis, and then it moves out of the nucleus via the nuclear pore and then goes to the ribosome where it attaches itself. So you get the marks, get all the marks. The reason why this book is changed in different color so that you know exactly where the marking point is. Yes. Then you're saying that another question is DNA based triplet code for serine. Serine is here, serine, but they are saying this, they are looking for DNA. So now we have to convert this into two times, because we have tRNA, we have to change it to messenger RNA, and then we go to DNA. So if it is tRNA, it is saying serine is UCG, so it's gonna be U goes with A, C goes with the G, and then G goes with the C. So it's gonna be TCG. So that is the code for DNA. So it means that you have to convert it into two times. They're saying that the first two amino acids coded by the molecule Z in the diagram from left to right. The first two amino acids coded by the Z and the diagram from left to right. So left to right. So I have this and I also have this. But I need to first read, this is messenger. I need to go to the table and check. So since it is transferring A, so I have to convert this into U goes with A goes with U, and then G. So and then another one is CAU. So AUG, I come here, I look for AUG, which is tyrosine, and then CAU, which is valine. What is the chance in the sequence of the nitrogenous base in DNA molecule called? What is the change in the sequence of nitrogenous bases in the DNA molecule called? This is gene mutation. So we call it gene mutation. Why? Because the bases have been changed. Then they're saying the code for this SCU on molecule Z changed to this. Explain the effect of this. This can also be answered by the book. If you go to the end of DNA, it is here. What will happen if this mutation changes the triplet 1 to this, or this to this? So basically, if you go here, then automatically it's being answered there. And then this is the permanent, is the mutation which is permanent alternation of DNA sequence that make up a gene. Question they have asked here, change in the sequence of nitrogenous base. How is it called? It is the gene mutation. So now, if this, we are saying that CUA, we are saying codon, so we have to change it into anticodons. So we are saying that CU is going to be GAA, and then this one's going to be GGA. If this codes for the same amino acid as this, there will be no change in the protein because it's just coded. It's like having two names, and I call you with the first name, you say yes, sir. I call you with the second name, still you say yes, sir. But if now I call you with a different name, you won't say yes, sir, because we're a different person. So if this codes for the same amino acid as this, the protein will remain the same. But if this codes for a different amino acid as from this, then it means that the protein will change. So it means that if you come here, GAA, it is leucine, while GAA, GGA is proline. It means that change of this to this, change of this to this will result in a different amino acid being coded from leucine to proline, which will result in a different protein. Change in CUU to CCU will result in a different amino acid coded. That is from leucine to proline. From leucine to proline, which will result in the structure of a protein. Yeah, I think I'm going to stop here. Yes, I'm going to stop here I'm going to stop here. When I come back, I will finish this paper from 2.3. Thank you very much. Please don't forget to subscribe. If you have many questions, drop them, or you can send them on WhatsApp. We will answer them before or with immediate effect. Thank you very much. Let's continue with the question 2.4. It's about genetics. We know that many students find challenges with the genetics. Question 2.4 from National Senior Certificate 2022, NCS 2022. The diagram below shows the inheritance of flower color in snapdragon plants. The two alleles controlling the flower color are red and white. It means that because the two alleles are capital letter, this is a capital letter, and also this is a capital letter, it means that both of them are dominant. So if both of them are dominant, here it's showing us that you have a dominant red crossing with white, and it gives you pink. When you have red and you have white, we produce pink. So pink is neither red nor white, but is an intermediate between the two. So the type of dominance which is there, the moment you get an intermediate, neither of the parent color produced in the offspring, it means that that kind of dominance is called incomplete. It means that red is neither dominant over white and white is neither dominant over red. An example is different presidents of different countries. When they sit on one table, all of them, they will talk about issues affecting them equally. So it means that neither of the president of country A nor the president of country B is dominant over the other. Why? Both of them or all of them are present. So neither red nor white is dominant over the other. So when they combine, they produce an intermediate. So let's go to the questions and we see. They are saying that state the type of dominance shown by the Snapdragon plants. So we have seen that it is incomplete dominance. If it was complete, then we would have got either red or white in the offsprings, or both. Red, some of the organisms are red and some of them are white. That is complete. If it was codominancy, we would have got a mixture of red and white. But now we get an intermediate, therefore it is incomplete dominance. Give a reason for your answer. The answer, we have explained it, that you produce an intermediate, or an intermediate color was produced. A gardener crossed two pink flowers. Use the genetic cross to show the ratio that is expected of when you cross two pink flowers. So it means that if both of them are dominant, definitely this is going to be capital R, capital W, capital W. Yes, they produce one gamete from this side and one gamete from that side. So this one is going to produce a capital R. This one is going to produce a capital W. So it means that this crosses with this, you produce this. And RW is an intermediate of red and white, which is pink. So it means that when you're talking about the genotype of pink, we have to use this. So what are you supposed to write? Okay. We are saying that now, the first thing you need to do is you say, because it is a second generation we are crossing, therefore it is advisable to use P2. So it's going to be P2. You tell us the P means parent. So P2, parent 2, because we have already parent 1. So now pink and pink, it means that that's P2. Yes, if it's a second generation, we use P2. P2, yes. We can say F2, second Fourier generation. We mark this as ones, like, so give you one tick there. So don't forget, if you write P2 or P1, F1, it must be, yes, it must be P2, F2, P1, F1. If you write one, you don't write the other one, then you get it wrong. For example, P2 and don't write F2, you don't get a tick. So now P2, you tell us the phenotype. The phenotype is pink. We cross it with another pink flower. The X must be like this. It must not be like this. No, the X must be like this. Then you go to genotype. What's the genotype? We have seen the genotype here. That is capital W, capital R, or capital R, capital W. Therefore, we're going to have to say here, it is capital R, capital W. Cross with capital R, capital W. Now, these must separate under meiosis. So you write the word meiosis. So meiosis, you form the gametes, capital R, capital W. Here, capital R, capital W. So we call these ones gametes. Gametes. Don't leave these gametes naked. In most cases, I always tell my students not to leave these gametes naked. They're not at the beach. All right. We circle them. We circle them. We circle them. Even if you don't circle them, still you get a tick. But I prefer to circle them. Why? So that we can differentiate them from the genotype. And always know that there must be a space between these two gametes. Don't combine them, because when you combine them, then it will look like it is a genotype. There must be spaces there. All right. Now these gametes, they're supposed to fertilize each other. Don't use the word fission. We won't give you a tick. Fertilization. Fertilization. When fertilization occurs, you can use a Punnett square, or you can use crosses. Since it is a monohybrid cross, you can use crosses. For example, this goes with that. So I'm going to have capital R, capital R, and now this with that. So it's going to be capital R, capital W. Now this with that is going to be capital R, capital W. Then this with that. So it's going to be capital W, capital W. Yes. So this is what we call fertilization. So these are the offsprings of the parents. So F1 offsprings. What are they? They said snapdragon. It means that those are the offsprings or the snapdragons. But what was the question? The question is saying, use the genetic cross to show the ratio of the expected phenotype in the offspring. So we are looking for the ratio of offspring. Okay, let's look at the ratio of the offspring. Now, capital R, it is red. So I have red. How many are they? It's 1. Comma. I have capital R, capital W. It is pink. Capital R, capital W is pink. So I have 2 pink. And then now I have capital W. It's white. I have 1 white. So basically I have 1, 2, 2, 2, 1. So that is it. So now, when you are looking for a mark, when we mark, we are looking for ticks, and then sometimes we put what called a compulsory mark. So in case we come here, we do this, we do, yes, this and that, we give you a tick. Phenotype, we give you a tick. It must have a cross. Genotype, we give you a tick. It must have a cross here. If it doesn't have a cross, the tick might not be given to you. So we come to the gametes, we give you a tick. And then we have for crossing, and you get the offsprings, we give you a tick. So because the question is saying give the ratio, so this one might be a compulsory mark. It might be a compulsory mark. So we give a tick, but we put the word C there, which means that it's compulsory. There is 1 mark short, which is meiosis and fertilization. Give you a tick. So how many marks are there? It's 1, 2, 3, 4, 5, 6, 7. And then in most cases, we give you 6 marks. And also sometimes cross, we can give you a tick there. So maybe we can say that this question is 6 marks. But how do we count? When you are counting, first count this. However much you get all of them, like you have all of this. Without this now, for this case, you won't get 6. You have to be with all of them. You have to be with all of them plus this. Or you have to be with this plus another 5 so that we make them to be 6. So that's how we mark the genetic cross. This question, it is 2022 Department of Basic Education, NCS. Another question is about evolution. Evolution. Here they're saying that diagram A and B show the ventral, that is the bottom side of the skull, of the skulls of 2 organisms. The diagrams are not drawn to scale. It means that they are not, that's not their size. So now asking, which diagram represents the skull of bipedal organism? Let me first explain the word skull. Skull. Some people, they confuse the word skull and cranium. Cranium is where the brain goes. The skull involves even the jaws. So when you are meaning where the brain goes, please don't use the word skull, use the word cranium. So skull is the whole thing, the head, including the jaws, the teeth, and everything. But when you talk about cranium, please, we are talking about where the brain goes. All right. Now, which diagram represents the skull of bipedal? Bipedal, it means that organism is walking on 2 limbs. If the organism is walking on 2 limbs, it means that what shows we have 3 factors which you can identify whether the organism is walking on 2 limbs or not. The first one is the position of foramen magnum. The second one is the shape of the spine, if it is S shape or C shape. S shape, it means that it allows flexibility for the organism, so therefore it is bipedal. And then C shape, the flexibility is not there, therefore it works on 4 limbs, quadrupedal. So the pelvic girdle for the case of bipedal must be short and wider to support the upper weight and allow easy movement. The other one, it must be small and narrow. Then foramen magnum for the bipedal organism, it must be in the forward position. That's how we want it, in the forward position. While the foramen magnum, it must be in the backward position for the case of the organism which works on 4 limbs. Why is it supposed to be in forward position? Forward position so that the spine can connect to the skull vertically. And then now the skull is going to face straight in front. Straight in front. Hence, the organism is going to walk on 2 limbs. So if you look at this, it's a little bit in the forward position. Don't say in the center. Center of what? No. Even this side can be center if I count from I measure from this side to this side. So it's going to become the center. So the center, we say in forward position. Well, this one is a little bit backward. So it is not in the forward position like this. So it is backward. So it means that this is a bipedal organism. This one is the quadrupedal organism. So they're saying, give one visible reason for your answer. So the answer here is B. If the answer is B there, therefore it means that give one reason. It's because the foramen magnum is in the forward position. So it is in the forward position. And then they're saying that tabulate 2 visible differences between the upper jaw. Now here, they're looking only and specifically the upper jaw. These jaws which you see. So don't talk about other parts. The upper jaw in diagrams A and B. When they say the word tabulate, that's a mark. The word tablet, even you can see that is 5 marks. So when you draw a table, please make sure that you draw a correct table so that you get all the marks. So you write here, for example, skull A and then skull B, for example. Make sure that you close the table. You lose marks here. Ne? Don't just draw a T and then you write. No. If you draw a table, make sure that you put the heading. Ne? Please close. Don't just draw it like that. Please close the table. Close the table. It doesn't cost you anything for you to close the table so that you don't lose a mark. Be neat when you are writing. All right, now let's compare. Skull A, skull A. This is skull A, skull B. So number one, they're saying the upper jaw. Let's look at these teeth. This and this. The canine here, they are big, meaning that they are well-developed canines. Here, the canines are less developed, meaning that they are small. Number two, this one has a rectangular palate. Here, it has a U palate. Ne? It's like a C or a U palate. Here, it is rectangular palate. So, those are some of the differences you can talk about because they are talking about the jaws. So the palate here is rectangular palate. Here it is U shape, or the jaw, they are U-shaped, and then here, yes. And then they are saying that so you get 2 marks for the difference and 1 mark for the table. Then they're saying explain the significance of the shape of the spine. I talked about it. That is associated. Shape of the spine, significance of the shape of the spine that is associated with the skull B. What is skull B? Skull B, we say that it is walking on 2 limbs, and the shape, if you look at it, it is S shape. Because they didn't talk about which shape is it, then mentioning the shape could make you to earn a mark. Therefore, the shape is S shape. Yes, S shape. But what is this S shape for? It allows flexibility of the organism. So it makes the movement a little bit easier. Then, in most cases, things related to the graph, I only point out what is very important and the most likely questions which are likely to come when they bring a graph, so that there is no cramming about the graph. So now, if they ask you to describe, when they say you're supposed to read the whole question. Herbicide is a chemical which kills a plant. Glyphosate is used to control the weeds in maize fields. The herbicide kills the weed, but it cannot kill the maize plant. The herbicide kills the weed, but it can also kill the maize plants. It has been found out that weed developed resistant to glyphosate treatment. Scientists carried out an investigation to determine the development of the glyphosate resistance in the weeds. The weeds were treated with the same concentration of the glyphosate each year from 2009 to 2016, and the percentage of the glyphosate resistant weed in the field was recorded every year. Okay. Results as follows. You see that glyphosate resistance, as year goes on, the resistance increases, but 2015 and 2016, the resistance became constant. So now I'm saying describe the change in the glyphosate resistance over a period of year. It means that here, whenever they ask you to describe, you tell us what is happening. Because if you draw this on the graph, not a bar graph, just a normal graph, you will see that it will be continuing, and then it forms something like this. So there is an increase from 2009, 2015. There is a steady increase, yes, in the resistance of the weed over the herbicide, and then it remained constant between 2015 to 2016. So it remained constant. So when you say description, you tell us what you see. Don't give us the reason why there is that increase. That is description. When they ask you to explain, then you tell us why, you tell us that there is an increase, and then you give us a reason why that increase is there. So that is what we call explanation. But if they ask you to describe in science or in here, you just tell us the shape of the what? Of the graph or the trend, how it moves. That's what they want. That's why you see that the marks even, they're not that a lot. Then they're saying that calculate the percentage increase in the glyphosate resistant weeds from 2010 to 2015. Show you're working. Here, whenever they ask you to calculate the percentage increase, whenever they ask you to calculate for the percentage increase, this is what you are supposed to do. Percentage increase is equal to the final percentage increase is equal to the final figure, the final value minus the initial value over the initial value times 100. Yeah, that's it. So in this case, they say that from 2010 it is 20, and the 2010 is the initial value, and then to 2015 is 65. So I'm gonna say the percentage increase is gonna be equal to 65 minus 20, yes, divided by because it's the initial, it is 20, then times 100%. So the answer you get is gonna be the percentage increase. So that's how we calculate the percentage increase. So this is a rough thing how you can answer questions when you really sit in this paper or any paper you're writing. You can really have no questions which are challenging. You are not going to see any new questions. All the questions you're gonna find in the paper are always the questions we ask you every day. Those are the questions you're gonna find in the paper. So there is no new questions because we don't have any new science which has come in. So the curriculum remains unchanged for a period of years. So no new questions. Don't expect new questions in your exam. So please try and focus on, we have the same format of setting, so we cannot set outside the examination guideline. So scientists isolated the gene from for glyphosate resistance from the weed and used it to genetically modify the maize plant. Okay, explain the economic benefit of making the maize plant resistant to glyphosate. Because the glyphosate, now look, they are saying that herbicide is a herbicide which kills plant. Maize is also a plant, and then we use it to control weed in the maize plantation or the fields. So we have to make sure that this glyphosate cannot kill the maize. So the reason why they make this maize to be resistant to the glyphosate, so that when you apply the glyphosate cannot kill the maize what? The maize plant. So that is the idea behind it. So I'm not saying that this is the exact question which is going to come, but when they give you such question and you don't have any idea, you can go back in the passage, most especially these questions which are involving of reading, go back, read again, and then identify. Then they're saying that draw a bar graph. Before I go to the bar graph, can you identify the independent variable here? You can identify the independent variable here. So is the weed dependent on the herbicide, or the herbicide depend on the weed? It means that the weed will depend on the herbicide over a period of years. That's why here you see that over a period of time or years. That's why you see here that glyphosate resistance, the resistance will accumulate based on the period of years, meaning that this herbicide will be resisted based on the period the weed has stayed in contact with the herbicide. Therefore, the resistance is depends on the contact time it has stayed with the herbicide. Hence, resistance becomes the dependent, and then the period of the contact becomes the independent. Okay, the bar graph. Now here, when you talk about the bar graph, you need to know the things we are looking for so that when you go to the exam, you don't miss any question concerning about the bar graph, concerning about the graph. The first thing you need to know is the caption. Sometimes some teachers don't explain, they just mark, they put a T, they put a C, they put a S. They don't explain this to their students. But what does it mean? How do you answer this question so that you don't skip any mark? All right. So now they're saying that the bar graph should illustrate the results of investigation in the first 4 years. So, okay. Now, I'm not going to draw the actual graph, but I'm just going to talk about how you can draw this bar graph without stress. So years are always an independent variable, meaning that this is time. Time is always an independent variable. So independent is always on x-axis. Independent is always on the x-axis. And then the dependent one is always on the y-axis. I always tell my students that if you forget which one is on the x-axis, which one is on the y-axis, you just write here the word ID, your ID. Ne? Yes, ID, which means that you know the ID you are using. Independent, dependent. So this one is going to go on the x-axis. Then you have put ID, X, Y. So this one is going to go on x-axis. This one is going to go on the y-axis. So that's how you can remember the x-axis and the y-axis. So you just do this small box. I repeat. Ne? You say ID, X, Y. So meaning that ID, independent variable on x-axis, dependent variable on y-axis. The first column on x-axis, the 2 or 3 or 1 goes to the y-axis. All right. Now, if you are drawing a bar graph, make sure that you draw a straight line. Don't mind about mine. Straight line. This is how we mark. You write a caption. Never forget about the caption. So we give you a tick for the caption. Then after that, you write the X, you write the Y here, you write the X-axis here. But when you're putting the value here, for example, in this case, they are saying yeah, and then the percentage, the glyphosate resistant weed percentage. So it means that the resistance is going to be here. Yes, the resistance is going to be here. And then units, it is in percentage. Then the time is going to be here, which is year. And then the years, they don't put anything. So if you just say time in years or just year, then it's fine. It means that now they're going to give you another tick for labeling the what? Labeling the scale. Ne? Not labeling the scale, labeling the axis. So if you label the axis X, they can give you a tick, or Y, they also can give you a tick, or they can combine the two. Now, you go to, after that, you go to the intervals. Please don't start with a 0 here, yet you want to start. If you come back here on the, you'll see that is 10, 20, 32. Okay, this one, it's fine. You can start with a 0. But if it is starting from maybe 100, and then you go to 120, you'll find out that some people, they start here 0, 100, 120, 140, 160. So the difference between 0 and 100 is not the same as from 100 to 110. So if you start from 0, make sure that, and you say a difference of 10, it must be 10, 20, 30, 40, 50, 60. It must be consistent. If you don't do that, you're going to lose a mark. And then now you're going to have to draw your bar graphs. It must be straight. Don't forget that these bars must be straight. It must be straight. It must be straight. Ne? So now the distance between these bars, I'm saying this is a rough distance between this space and this space must be equal. It might not be exactly the same because you don't use a ruler when you're marking. No, but when you look at with your eyes, you see that these distances are the same. So it must be equal. These bars, the size must also be equal. So that we give you, if the scale, the interval is the same, we give you a mark for the scale. Yes. If you have drawn the bar graph, not a histogram whereby the bars are this connected to each other, then we give you correct type of the graph. We give you a correct type of the graph. We give you the mark for the caption. Then we give you a mark for labeling the axes. We talked about it. Then we give you a mark, maybe 3 of them. We give you a mark for plotting if you have plotted this correctly. So that's how we mark the bar graph or any other graph of any kind of nature. So basically, that's how we mark. Then the last question is natural selection and also out of Africa hypothesis. Still here, I'm not going to answer the actual, like the way it is here. I'm just giving you the ideas how you can answer these questions without stress. Because when you are bringing the question, we can bring it in a different version. Yes, we can bring it in a different version so that you don't lose those marks. So when you modify, where do you modify and how do you modify it? A population of lizards on the island A were well situated to feed mainly on insects. So it means that these were feeding on insects. Scientists moved the 5 adult pair of the lizards species to the neighboring islands. And they are saying the island A, island B, here they reproduced and a new population formed. They're saying the island B has large supply of plant with the fibrous leaves and fewer insects. It means that now the food has become scarce. So this one is going to become a problem. Yes, exposure to this insect new environment may have caused the lizard to undergo evolution. Okay, they're saying 63 years later, scientists returned to the island B to conduct a further investigation on the lizard population there. They observed that the jaw size of the lizards had increased, and scientists also analyzed that the stomach content of the lizard found that it was mainly plant-based. They confirmed that the 2 populations still belong to the same species. They confirmed that the 2 populations still belong to the same species. Describe how scientists could confirm that describe how scientists could confirm that there was a change in the jaw size between the lizards of island A and the lizards of island B. So the question might not come like this, explaining this, but it can come in a different version. So we shall go and then see exactly where we need to have the answers from. So basically, what the scientists could use to confirm or could confirm that there was a change in the jaw is, this is an obvious answer. It means that they had to measure the lizards on the island A and the lizards on island B, and they check whether the jaw size is the same, and they can compare the differences if the difference is there. Then explain how the large jaw of the lizards on island B would be structurally suited to eat the tough fibrous. Explain how the large jaw of the lizard on island B would be structurally suited to eat the tough fibrous leaves. Yeah, this could be about the jaws being strong, meaning that the muscles are strong enough, and also the teeth, so that they can have a strong bite, and then they can chew that food. It means that the muscles, to do that, it must be strong, so that the force of grip is high, and then they are able to chew this kind of what? Kind of food. How did the scientists determine that the two populations of the lizards, both on island, still belong to the same species? Don't forget that a population, a species, these are organisms that can interbreed to produce a fertile offspring. So if they have the two populations and they try to allow them interbreed, and they produce a fertile offspring, therefore they can confirm that these are still the same species. But if they cannot interbreed to produce a fertile offspring, then automatically they are not the same species. But if they could interbreed and produce a fertile offspring, therefore it means that they are still of the same species. Explain the possible effect that evolution of the lizard has on the biodiversity. Explain the possible effect that evolution of the lizards has on the biodiversity. Here, it could be, it can increase the biodiversity or it can reduce the biodiversity. So basically, those are the two ways. If it forms new species, then automatically it has increased the biodiversity. If it takes away an existing species, then it forms another species, then it means that it is decreasing the biodiversity. However much it's creating, but it's decreasing because the other one is no longer existing. Or it could be 3 species, and then because now the food has come in a different angle, the 3 species, they modify into 1 species, or they modify and the other ones, they become extinct. They become extinct, meaning that now they have also decreased the biodiversity. Use the Darwin theory of natural selection to explain the evolution of lizards with large jaws. Use the Darwin theory of natural selection to explain the evolution of lizards with large jaws. Here, the principle is the same. The principle of natural selection, whatever you explain, use the same principle. The first point you have to talk about is variation. There was variation among, now you have to tell us what they gave you, among the lizards, those with the large jaws and those with the smaller jaws. Because the jaws are used to bite or to eat this tough food, therefore, those lizards with big jaws, they could eat enough food, tough food, therefore they survived and reproduced in large number. Those with the small jaws, they could not eat enough food, therefore they died in large number. And this is called natural selection. Yeah, previously we are giving a tick on natural selection. This is called natural selection, but the chances are high that we're no longer giving this marking point of this is called natural selection. But put it there, you never know that maybe we can review about it. This is called natural selection. So those with big jaws, they pass their genes to the next generation because they reproduce in large number. After many generations, all the lizards have big jaws. There is variation, I'm repeating, there is variation among the lizards. Those with, you tell us the variation. Those lizards with large jaws and those with the small jaws. Those with the large jaws, they could eat the tough food. Yes. And then they survived, they reproduced in large numbers, they passed their genes to the next generation. While those ones with the small jaws, they could not survive, they could not eat enough food, therefore they died in large numbers, and this is called natural selection. Those which survived, they passed their genes to the next generation, and after many generations, all the population of the lizards were made up of large jaws. That's how you can explain using Darwin's theory of natural selection to show how this evolution of large jaws came about. Lastly, on this paper, please, you should never go to the paper without knowing these questions. The out of Africa hypothesis. which states that modern humans originated from Africa and migrated to the rest of the world. Don't forget the word modern. Modern. However much they might not give it as a tick marking point, but in most cases, we criticize when the word modern is not there, because not all humans are modern. Homo habilis were humans. That's why you call it homo, which means a man. Homo erectus were humans. That's why it's called homo, is a man. Homo sapiens is a human. That's why you call it homo, which means a man. So Homo habilis, the upright, the handyman. Homo erectus, the upright walking man. Homo sapien, the wise man. Those are, all of them are humans. That's why we are referring to modern humans. So please put the word modern humans. Homo sapiens, which means the wise man. I don't mean the school, I mean the man, you who is listening. Someone's going to ask me, what about us who are ladies? Yeah, is a man in science. All right. They're saying that name the family to which modern humans belong. They belong to a family called Hominidae. Hominidae. So they belong to a family called Hominidae. Yes, that's the family to which it belongs. They're saying that which genetic evidence is used to support the out of Africa hypothesis. We have two genetic evidences, but we only teach one. We have what called mitochondrial DNA and Y chromosome. But now we don't talk about the we don't talk about the Y chromosome. Only now the syllabus or the curriculum is talking about the mitochondrial DNA. So the evidence we give you here is mitochondrial DNA. Describe how fossil evidence is used to support out of Africa hypothesis. I've been telling these kids that these are the same questions we ask you. And if you check the book, our book we use, you can download even yourself. That's exactly how we mark exactly. This is the same way how they ask you this question. This book was released a long time ago before even these papers are being said. All right, let me answer. You have to talk about the oldest fossils which were discovered in Africa. For example, you have to talk about that Adipithecus remedus was discovered in Africa. You have to talk about that Australopithecus. Among Australopithecus species, we have Lucy, that is Afarensis. We have Taum child, that's Africanus. Mrs. Place, Africanus. Little fruit, Africanus. Carabo, Sediba. All those Australopithecus species were discovered in Africa. Homo habilis, also in itself, was discovered in Africa. That is the handyman. Now, when you reach here, you add the word the oldest. The oldest species, the oldest fossil of Homo erectus and Homo sapien we also discovered in Africa. That's what you want. So you may not talk about all the Australopithecus species, all of them. You can just talk about one or two. So Adipithecus discovered in Africa. Australopithecus species discovered in Africa. For example, Taum child, Mrs. Place. Then you'd go to Homo habilis, discovered in Africa. Homo erectus, the oldest, Homo erectus and Homo sapien, discovered in Africa. Let me show you what I'm trying to say. This is what I'm trying to say. Evidence to support out of Africa hypothesis. There are three: the mitochondrial DNA, the cultural evidence, and the fossil evidence. This is what I'm trying to say. That fossils of Adipithecus were only found in Africa. Fossils of Australopithecus, e.g. Carabo, Little Fruit, Taum child, Mrs. Place, all were found in Africa. Homo habilis were only found in Africa. Oldest fossil of Homo erectus and Homo sapiens were discovered in Africa. This shows that modern humans originated from Africa and migrated to the rest of the world. You see that even I highlighted the key points which you need to know when you're writing in your exam. Yes. So as I'm winding up, I will recommend you to use this book. Go to our website, download it free of charge. If you want to be printed, that's a different story. But go use these resources. I took time to write this book, and I wrote it with the care so that every child of this country in South Africa must pass. You don't have a right to fail. You have a right to pass. You must demand for your rights. What is your right? The right of passing. Please, never ever demand for the right of failure, because you have the ability for you to pass, not the ability for you to fail. I wish you the best. Make sure that you go through these papers. You will make it. Thank you very much. Don't forget to subscribe, like, and share to others so that they also benefit in this before they write their final exam. Thank you very much. M.Said as usual. Welcome back to our session, which is guiding you for paper 2. We'll be looking at questions, most likely examinable questions, which are going to come in your paper 2, both in prelims and final exams. Don't forget that in most cases, we don't set new questions, but we use the questions which are prevailing, and then we modify them. So I'll be showing you how to answer these questions, and I'll show you different questions. Some of them I'll be just speaking about them, and then how do you answer those questions. So keep on so that you don't miss out anything, and don't forget to subscribe, like, and share to others so that you can also benefit. M.Said as usual. Let's start with the question 3. The diagram represents the portion of nucleic acids. Before I start this, don't forget that we have specific topics which we are supposed to look at in paper 2. The first topic is DNA, code of life. The second topic is meiosis. The third topic is genetics. The fourth topic is evolution, but both evolution in natural selection and then human evolution. So it means that these are the topics which are being set in paper 2. So when you are reading, you must focus on these 4 topics or 5 topics. So we're going to look at all questions, and then the most likely examinable questions or questions you're most likely to find in your paper. If they bring a diagram like this, you have to find out which diagram is representing. So this diagram represents DNA. Why DNA? Because of this, the weak hydrogen bonds. And then it's two-stranded. What are some of the questions which are most likely to be asked about the structure of DNA? Apart from labeling the structure of DNA, they can ask you to describe the structure. Apart from labeling it, describe the structure of DNA, and they put there 6 marks, whereby you have to tell us DNA is made up of nucleotides. Each nucleotide is made up of a phosphate, a nitrogenous base, a phosphate, and a nitrogenous base, a sugar, and a phosphate. It forms 2 strands. Each strand forms 2 strands. Each strand is joined together. So you describe the characteristics of DNA. Basically, what DNA has. It forms 2 strands which twist around to form a double helix. So you can get the 6 marks from there. So in this case, let's label it and then find out what is a V. What is V? V is the nucleotide, because a nucleotide is a nitrogenous base, a sugar, and a phosphate. So this is a sugar, and then nitrogenous base, and then a phosphate. So this circle or box is showing you a nucleotide. Then Z is weak hydrogen bond. Use the word weak. Weak. We want to see the word weak. Then hydrogen bonds. Weak hydrogen bond. And then this is a nitrogenous base. Nitrogenous base. And then also this is a phosphate. Sorry, a nitrogenous a sugar molecule, which is in this case it is ribose, deoxyribose sugar. And then Y is a phosphate group, as we saw it here. So now, since you know that this is a structure and you know how to label it, now let's talk about name the nucleic acid which is being represented. So we have seen it that this is DNA. And they're saying name 2 places in animal cell where you can find this DNA. Where can you find this DNA in animal? You can find in animal in 2 places. That is in the nucleus, and then in mitochondrion. If it's one nuclei, if it's one mitochondrion, if there are many mitochondria, if there are many nuclei, if it's one, it's called nucleus. So it should not confuse you. They are saying name portion V. They are looking for portion V. What is portion V? We say that these are the building blocks. This is the building block of DNA. So it is the nucleo. Molecule Y. Where is Y? Molecule Y is a phosphate. We have seen it that it is a phosphate group. Then they are saying that bond Z. What is bond Z? Bond Z is weak hydrogen bonds. So I think we have seen the answer here. Weak hydrogen bond, we have seen here. Phosphate group, nitrogenous base, we have seen it. And then portion W, which is the nucleic acid. Sorry, which is the nucleotide. What is the natural shape? When they say natural shape, many students, they say helical, double helical. No, the only answer we accept in exam is double helix. Nothing else. You write something different from that, we won't give you a tick. Name the process during which this molecule makes exact copy of itself. How does DNA make exact copy of itself? How does it replicate? So the answer is DNA replication. So that is the process by which DNA makes exact copy of itself. Why does it occur? Some questions can come here. Why does DNA replication occurs? It occurs to increase or to double the chromosome number. To double the chromosome number. Where does it occur? When you talk about where does it occur, then you talk about the nucleus. And then when does it occur? It occurs during interphase. Yes. And then why does it occur? Why does it occur? We say that to double the chromosome number. To double the chromosome number. And then lastly, how does it occur? That is the process of DNA replication. If they bring a question like this, how do you answer? This is a DNA molecule. So if you look at it here, you see it is a DNA molecule. And why is it DNA molecule? Because of the thymine, which is indicated in the nitrogenous base. And then also the weak hydrogen bonds. two strands, so it identifies it to be a DNA molecule. And it cannot be RNA, because even here they say that this is what? This is a DNA replication. Yeah, so question them. Molecule W, where is molecule W? We have seen it, molecule W is here. It is a nucleotide, correct. Molecule U, where is molecule U? The whole molecule is DNA, correct. Then they are saying that Z is weak hydrogen bonds, correct. And then what is V? How do we know the nitrogenous base V? Because nitrogenous base is compatible to this or complementary to this, therefore, and this is given it here, which is thymine, therefore V is equal to A, which is adenine. And then they are saying that name the part of molecule W is W, X, and Y. X is the phosphate group, correct? And then Y is the oxyribose sugar. Why don't I say sugar? You have to be specific because this is DNA, so it is the oxyribose sugar. Where in the cell does this process take place? Where do we find? We have answered this question that this process occurs in the nucleus. In the nucleus. And then they're saying that name the phase of cell cycle where replication takes place. We have already answered it, and then we say that interphase. So at least when they bring such questions, you must be able to answer accordingly. So first try to answer what is being labeled. The moment you know what the picture depicts or what a picture represents, at least you are able to bring your mind and then you concentrate in that area. I'm not going to spend too much time here, only maybe if I see a very difficult question there, then that's when I can try to answer related to this, because I wanted to go to transcription and the translation. But molecule C is phosphate, and then this is a nitrogenous base, and then this is phosphate, sugar, deoxyribose sugar, and then nitrogenous base. It forms what you call a nucleotide. And then weak hydrogen bonds, and then this is a sugar, this is a complementary base. So this is a complementary base. And then here, yes, they say this weak hydrogen bond. And then you have this. How do you call that? That is not a strand. We call it a backbone. And then the whole strand must include with the nitrogenous base and this. So the difference between a whole strand and a backbone is the backbone does not involve the nitrogenous bases. If you are asked what is this bond, this is a phosphate sugar bond, but it is a covalent bond. If you refer it to physical science. Okay. Two scientists who discovered, James Watson and Francis Crick, name one organelle where DNA is found in the nucleus and the mitochondria. Describe how mutation of DNA may change the structure of the protein. The moment DNA molecule changes, is going to change the sequence of the bases on messenger. And the messenger RNA is going to change the sequence of the amino acids in which they are being brought to form a protein. So if a different protein is formed, if the amino acid leads to a different sequence, then a different protein is going to be changed and or is going to be formed. Then amino acid leads to the same sequence as previous one. Then the protein is not going to, a new protein is not going to be formed. Table 2 different structure difference. Now, if the major point here is structure, the word is structure between the monomer and they are saying about the monomer of DNA and RNA. So here they are talking about the monomer, and if you look at the monomer, they are looking at the nucleotide of DNA and the nucleotide of RNA. So you only look at the structure, which is the nucleotide, of which the nucleotide is made up of a phosphate, a sugar, and then the nitrogenous base. So there are only two differences here you can look at. You can look at the sugar. DNA nucleotide has deoxyribose, deoxyribose sugar, while RNA has ribose sugar. And then DNA has thymine, while RNA has uracil. So after that, you draw a table. It must be a complete table. Yes, it must be a complete table. And then you put DNA, and then you put RNA at least. Then you get a tick here, a tick here, a tick here, a tick here, and a tick for the table, which makes it to be 5 marks. And if you find a question like this and tablet, and there is only 10 minutes or 10 seconds remaining, don't waste time. At least you can grab that 1 mark. Just draw a table like this, and then put DNA, and then put RNA. Yeah, even if you don't have anything here, then we shall come and give you a tick. At least you will score that 1 tick. So yeah, please try to use different ways in which you can get marks. Yes. Yeah. So this is the same thing we have been trying to discuss. Phosphate, you have seen them, and then M2 process, which require the strand of the nucleotides in a single strand of DNA. Sometimes people think that DNA can be a single strand. It can be a single strand. What makes this one to be DNA is because of the thymine, which is being indicated there. So thymine is in this form, while adenine is in that form. Then they're saying that how many nucleotides are represented in the diagram? Yes, let's try to answer this. How many nucleotides? A nucleotide is a phosphate, nitrogenous base, and a sugar. So this is 1 nucleotide, it's the second nucleotide, it's the third nucleotide, and then this is the 4th nucleotide. So the answer is going to be 4 nucleotides. And then they're saying phosphate molecule, then phosphate molecules, you just count them 1, 2, 3, 4. So the answer is also 4. And then they're saying that how many nucleotides, okay, I done with that. Sugar molecule, there are 4, but sugar, this is the phosphate, and then this is the sugar. Write down the nitrogenous base from top to bottom as indicated by the arrow as complementary base strand and this molecule. Here is just T, they're saying on DNA. Complementary. So they are not talking about convert it into RNA. Don't forget that A goes with the T, then C goes with G. So whenever there is A, T, you put A. Whenever there is C, you put G. Whenever there is A, you put T. Whenever there is C, you put G. So basically, that's how it's supposed to be answered. Name 2 processes that require the DNA strands of DNA molecule to separate into single strand as shown in the diagram. Which processes? 2 processes. We only find this in when the DNA replication is going to take place, and also we find this when there is a transcription. Remember here you form 2 strands, 2 separate strands. Each strand is acting as a template. Well, here you form 2 separate strands. Only one strand will act as a template. And while here 2 strands will act as a template. So these are the 2 processes which make DNA molecule to open. DNA profiling is also not a big deal. Technique, you have to know how to identify this. Read the question very well and then find out. Don't just answer the question. Detectives were investigating crime scene, found the blood on the broken window. Then they suspect that the blood was that of the criminal. To identify the criminal, they analyzed the DNA sample from the blood and compare it to the 4 suspects. Now, look, this is DNA sample, and then these are the suspects. It means that these are the DNA they found on the, but you have to see where it is corresponding. This one goes with that. This one goes with that. Still, yeah. This one is that. Use a ruler to identify this. That this with this. This with this. This with this. This with this. So it means that Jenny is a suspect for that case, whose DNA profile is matching with the DNA sample obtained from the crime scene. Name the technique which was used. It's DNA profiling technique is DNA profiling, and what is being shown here is a DNA profile. So you must know the difference. DNA profile is not a technique. DNA profile is what you obtain when you analyze the DNA sample. So who was the possible criminal? We have seen it is Jenny, because the DNA profile, or the profile, the DNA profile of bars, there of sample, DNA sample profile is matching with the profile of Jenny, or the band of DNA profile sample, they are matching with that of Jenny. So set one other use of DNA technique, or state other use of the technique identified in the in the question above, meaning that the DNA profiling, what is it used? Can be used to identify the relatives. They have talked about the criminalism, so you don't talk about that anymore. So talk about the identification of the perfect organ donor. You can talk about lost relatives. You can talk about diagnosis of the inherited diseases. So there are so many uses you can use to when you are dealing with the DNA profiling. So it's the same thing. You see that is the same thing. I don't usually take too much time here because these questions are really open. Who is a suspect? You know how we have identified it. What diagram is DNA profile? What does the diagrams above represent? They're not saying the technique, they're asking for the diagram. Therefore, it is DNA profile, not DNA profiling, because here they said which technique, name the technique. So it's DNA profiling, but here it is DNA profile. Which suspect? Look at the bands and then you match them, give a reason. So the suspect, if you look at them, you'll find out that Q is most likely to be the suspect, because the bands of broken glass, they are matching with that of Q. So they're saying that name 2 possible disadvantages of using this evidence in the court. It might not be accurate 100%, because number one, they can human error, number one, and also it may not be unique if a small sample is used. Also, they can, a human being can manipulate them, so it may not be 100% accurate. So we have seen many questions concerning about DNA profiling. Download this and then try to do these questions. Where you get stuck, come back to me. Ask me questions. I'll be able to help you out. My number is always on the website, 084-833-1738. Yes, and then speak to M.Said, who is always ready to help you out. Then another question is still the same tablet. Whenever you talk about tablet, make sure that you draw a nice table, and then so that you get that 1 mark. That's why there are 5, because they're asking for 2. They give you 2 marks there, and then 1 mark for tabulating. Describe the process that is being represented or responsible for formation of molecule B. So here, molecule B is messenger RNA. Why messenger RNA? It's because of this. So this is not, you see, this is messenger RNA, which is uracil. Therefore, and also one strand is being used. So that's evidence to show that this is a transcription, because they are saying this is non-coding strand. So it means that only this is acting as a template. Yes. So they are saying that describe. So what are they looking for here? Describe the process that is responsible for formation of B. If you look at B, is messenger. So you describe. How do you describe here? Let me just talk about it. As I told you, talk about DNA double helix, unwind, weak hydrogen bond, break, and unzip. So when the weak hydrogen bond break, it's the same thing saying unzip. So you get 1 mark there. To form two separate, the word separate must be there, two separate strands. Only one, you see it here, that only one is acting as a what? Is acting as a template. Only one is acting as a template. So it's being shown there that this one is acting as a template, and then using free floating messenger RNA. So you have to write messenger RNA nucleotide, yes, to form a complementary strand where adenine goes with uracil, and then guanine goes with cytosine. Adenine goes with uracil, and then guanine goes with cytosine. The complementary strand move out of the nucleus via the nucleopore to the cytoplasm on the ribosome. So basically, that's what you're supposed to explain. Please don't try to bridge it, because it's a process. One step leads to the other. If you start with what is on at the bottom and then you start with it, then you lose marks because the sequence is not the same. So you cannot start with the last step and then you end with the first step. You have to start with the first step and then you end with the yes. So molecule B, we said it is messenger RNA. Name the process is transcription. Explain how the protein molecule would be affected if codon GUU is changed to CGC. So if it here, it depends on two things. If there is a table, yes, now you compare the table and see if it's gonna lead to a different amino acid. But because now this is open, then we assume that it's gonna be a different amino acid. For example, if you say that you started with GUU, yes, and then GUC, from this to that. If this, let me explain this. If this is tryptophan, yes, codes for tryptophan, and this one also codes for tryptophan. However much this has been changed to this, is going to code for the same amino acid. Therefore, the functioning of the protein is not going to change. But if this tryptophan, and then now this one codes for example, leucine, then it means that there will be a change in the functioning of the protein, because this is tryptophan and then this is leucine. Therefore, a different protein will be changed, will be formed, and then still the functioning of the protein will also change. So explain how the protein molecule would be affected if this is changed to this. So therefore, you tell us that if GUC is changed to GUC, GUU is changed to GUC, because they say that codon this changed to, because this, when they say codon, it means that we have started from the messenger RNA. Then this will lead to different sequence of amino acid. Different amino acid will be brought to the molecule or to the messenger RNA, not messenger RNA, will be brought to different amino acids will be brought to ribosome, and then a different protein will be formed. So that's basically you have to understand. And then when you come here, this is translation, because it is occurring on the ribosome. So what is A? A is messenger RNA. What is B is the ribosome. And then what is C? C is the transfer RNA. What is Z? Z is the amino acid. So this is the monomers amino acids, monomers of the protein. And then this is the transfer RNA, you have seen it, and then this is the ribosome, and then this is the messenger RNA. So basically, you need to do that. And then what is this? These are 3 bases, so therefore these are codons. Yeah, remember, the 3 bases on the DNA, we call them codes. And then 3 bases on the messenger, we call them codons. And then 3 bases on the transfer, we call them anticodons. Organelle X, what is organelle X? We have seen it. We have seen all the questions. You see now, when you start with trying to label them, you'll be able to answer all the questions. Molecule W formed, you see, organelle Z, X, we have seen it, ribosome, monomer W, the monomer. They're saying the monomer of molecule W. Remember the building blocks, that's what we call the monomer. Therefore, it's going to be the nucleotide. Be the nucleotide. Yes. And then molecule W and Y, W and Y, we have seen it. W is messenger RNA, and then Y is the transfer RNA, and then molecule organelle X, we have seen it is the ribosome. So still the same thing. You can see some people, they call this a protein. This is not a protein. It is only a protein if it is 50 and above amino acids. So what is this then? This is a polypeptide chain. Yes, so it is a polypeptide chain. So what is X? This is the amino acid. And then what is this? This is transfer RNA. And then what is Z? We have seen them. These are anticodons, anticodon. And then this is messenger RNA, and the 3 bases on the messenger RNA, we say we call them codons. You see, you can't fail such questions. When you come to the question, you see that they're asking you the exactly same thing, and then asking you describe the process of transcription, which we have described. So basically, at least you have some light on DNA, and then maybe we will talk about the process of translation, and then after that we wind up with the coding and then the mutation. These are some of the short question which you need to know before you go for the exam. Number 1, DNA replication. You have to know DNA replication, the process by which DNA makes the exact copy of itself. Where does it occur? We have seen it in the nucleus. When does it occur? We say that it occurs during interphase. And then we said, why does it occur? We say that to double the chromosome number. And then how does it occur? That's the process of DNA replication. And don't forget that here you have to explain that here the you form 2 separate strands, and each strand is acting as a template, and that is the major difference between DNA replication and transcription. So in most cases, people confuse transcription and the DNA replication. Then number 2, describe the location and the structure of DNA. Here I explained it at the beginning, the structure of DNA. In most cases, give it 6 marks. And then location, we have 3 locations, that is nuclear DNA, mitochondrial DNA, and chloroplastic DNA. So if they talk about in plants, then you talk about all of the 3. But if they talk about in animals, you only talk about the 2, that is the, because we don't have a chloroplast in animals. So describe the structure of RNA. Structure of RNA, it's a little bit the same as DNA, but only few things which are not there. That is the thymine, the double strand, and then also the weak hydrogen bonds. Describe the involvement of different types of messenger RNA process in protein synthesis. Here they are talking about the functions of each. For example, you talk about transcription and then translation. So it is like you are describing the whole process of DNA of protein synthesis. Another thing you need to know here is also describe transcription in detail, and then also translation also in, yes. And then also what they can ask you is why is the mutations which occur. And I say that if you change the code, for example, from GUA to GCA, then if these two they code for the same amino acid, therefore the protein will not change. The structure of the protein will not change. But if the different amino acids are being coded for, then the structure of the protein will be changed. So let's go to meiosis. Let's go to meiosis. There are so many questions which can talk about DNA. And also the base calculation, whereby we're supposed to talk about if adenine is equal to 10%, what is the percentage of guanine, or the ratio of guanine, whereby you have to tell us that adenine goes with the thymine, and then cytosine goes with the guanine, whereby if this is 10, or definitely this is 10, and then 10 plus 10 is equal to 20. Since this is percentage, therefore I'm going to say 100 minus 20, which means 80%. And then 80%. So what happens? So it's going to give us 80% for these two. So divide by 2. And then from 40%, therefore this is 40. Then this is 40. So this is just a simple recall. I hope you remember. Please don't go without knowing it, because at least those 2 marks you'll be able to answer them. And then if they are looking for the ratio, you can put them maybe adenine now. Adenine, we said it is 10, and then maybe with the guanine we said is 40. So divide by 10, and then we're gonna get 1, 2, 4. So sometimes they swap it and they say 4 to 1, and then because when now you have seen 1, and then you have seen 4, you have seen 4, you have seen 1, you just circle that. It is what? It is wrong. So when you go to meiosis, so many point where crossing over takes place. We call it chiasma. You see a chiasma. Splitting of the cytoplasm, we call it cytokinesis. Okay, we can answer them. Failure of the chromosome to separate during meiosis, the non-disjunction, the structure in the cell that forms spindle fiber, centrioles, and then the phase of meiosis, where homologous chromosomes are aligned along the equator, that is Mera phase 1, because they're saying homologous chromosomes. And the point at which the 2 chromatids of the chromosome join, we call it centromere. And this division of the nucleus, is karyokinesis. And then they're saying that the site of meiosis in a female, because in female we find meiosis in ovary. So you write ovary there, ovary, and then the exchange of genetic material between chromatids in the homologous chromosome, which is crossing over. And then they are saying the genetic disorder caused by extra chromosome on chromosome number 21, that is Down's syndrome. Yes. And then they are saying that the structure formed between the centrioles by the centrioles during the cell division with spindle fibers. Yeah, spindle fibers. And then they're saying that non-sex chromosomes in human, we call them non-sex autosomes. And then they're saying the condition in the cell where there is only one set of chromosome. How do you call that condition? We call that condition haploid. haploid. And then the structure that is responsible for formation of the spindle fiber during spindle fiber, we say that is called the centrioles. Yes, and then the A chromosome aligned along the equator, none, because here it must be metaphase. Random arrangement of chromosomes, it must be metaphase. So it's A, B only. The site of meiosis in plants, anthers, and ovary, both in, this is male, this is female, and then the process of meiosis produces meiosis result in 4 haploid daughter cells, 4 diploid. So this one becomes wrong. This one becomes wrong. So this is the correct one. The mechanism to introduce genetic variation, crossing over, is correct. Random arrangement of chromosome is also correct. So it is both A and B. So basically, you see, yeah, so now we are starting the other part of meiosis. All right. Please don't forget to subscribe, stay in touch. If you are in any problem, we need to smash these questions, and then we get all the marks at the end of this period. Yeah, when you come to meiosis, there are some questions we can talk about. Let's see what we can go through so that they can also benefit you in answering questions related to meiosis. I know it is a simple topic, but is a little bit challenging to some other. But let's see if we can smash these questions, and then we get a better understanding. If you don't understand the topic, let me know. I'll help you out so that at least we can have more distinctions in this year. All right. They are saying that here you are seeing diagrams. What comes into your mind is the kind of phases which this diagram is indicating. So, what is C? C is spindle fibers. What is B? This because there are 2 chromosomes of the same kind or the same size and structure, therefore is homologous chromosome. Then the joint between 2, or the joint between 2 chromatids of the same chromosome, it is a centromere. And then diagram A, what does diagram A represent? Because of this one is starting to form, so it can represent prophase. Prophase 2. Why prophase 2? Why not prophase 1? It's because now this they are individual, they are not in pairs. And then number 2, you see that crossing over took place a long time ago. And then this one is metaphase. The chromosomes are aligned. If you turn this, and then comes this side, and then this goes that side, so that this thing is in between your chest. So you see that if this is one side of the chest, and then this is on the other side of the chest, I think now you see what I'm trying to say. So you see that they're in the middle. So why? These are the spindle fibers. Yes, if this is the spindle fiber, if this is the spindle fiber, and this is the spindle, centrio. So these ones are in the middle. That's why you say that it is under metaphase. But metaphase, which metaphase? It is metaphase 1, because these chromosomes are in the middle. Then they say identify A. We have seen it. A is centromere. B, what is B? B, we have seen it homologous chromosomes. Then C, we have seen it as spindle fibers. Identify the phases in diagram 3. Diagram 3, because they are separating. Yes. So if they are separating, then they are moving to opposite poles. You see them? They are moving to opposite poles. Therefore, it is anaphase. But which is it? Anaphase 1 or it is anaphase 2? Is it anaphase 1 or anaphase 2? Because now there are chromatids which are moving to opposite poles. Chromatids moving to opposite poles. Therefore, it's going to be anaphase 2. anaphase 2. And then they're saying that if it was anaphase 1, then this would have been individual chromosome. This would have been a chromosome moving to the other side. Also, this one would have been a chromosome and a centromere there. This would have been a hollow chromosome, and also this would have been a chromosome. So it would have been anaphase 1. But because their chromatid is moving to opposite poles, therefore it is anaphase 2. Then they're saying that write down the number of the diagram shown in the sequence which the phase occurs. The number of the sequence. They're saying write down the numbers of write down the numbers of the diagram to show the sequence in which. So basically, we have to look for meiosis 1. The chromosomes are supposed to be in homologous pair. So this one becomes, this is homologous pair. So it is 1. Therefore, it is what? It is metaphase 1. And then this one is prophase 2. And then after prophase 2, they separate, which is, so it's going to be 1, it's going to be 2, and then it's going to be 3. So when you are writing the numbers here, it's going to be diagram 2, diagram 1, and then diagram 3. So it's going to be diagram 2, diagram 1, and then diagram 3. Then one difference between metaphase 1 and metaphase 2. In metaphase 1, the chromosomes are aligned along the equator in homologous pairs, while in metaphase 2, chromosomes are aligned along the equator singly. They are individual chromosomes are the one which align along the equator. So that is the major difference. And then if we see another question, so label it. Very simple. You see, we usually bring the same kind of questions. So this is the spindle fibers. This is the centromere. This is chromosome. Non-homologous chromosome is 1 chromosome. And then this is the centriole, which is being produced by centrosome. Okay, centriole. And then what causes these differences in the chromosomes or chromatids is because of crossing over. When does crossing over takes place? It takes place during prophase 1. How does it take place? Homologous chromosomes, they align together. Chromatids of the homologous chromosome, they cross over at a point called chiasma, and then they exchange the genetic material, and then they separate to form different chromosomes which are genetically different. Basically, that is the process of crossing over. You can't go to the exam without knowing the process of crossing over. Identify the phase of meiosis in diagram 1. Diagram 1, because now, yes, the chromatids are separating, is the same thing like this one. It's the same thing. The same thing like this is exactly the same. Yes. So it's gonna be anaphase 2. And then this one is going to be metaphase 2 because the chromosomes are aligned along the equator singly. Yes, they align along the equator singly. Then they're saying that identify the phase. You are done with that. State what happens to structure D in the next phase. What is structure D? Structure D, these are the chromosomes. So the chromatids, they move to opposite pole due to contraction of the spindle fiber or due to the shortening of the spindle fiber. Name the process during which genetic material were exchanged. What was exchanged? We say that the process is called crossing over. And then we said it crossing over. And then we said it occurs during prophase 1. State the consequence if the mentioned does not occur. If this process does not occur, it means that organisms, it will reduce what you call the genetic variation. It means that more organisms will be more similar to each other. So it will reduce variation among the organisms because crossing over leads to genetic variation. Then they're saying that give the number of the chromosome present in the original parent cell in this diagram. They didn't say this is a human cell. I don't see anywhere there saying this is a human cell. So you have to know, you have to use the number which is being given. So because this is meiosis 2, and meiosis 2 leads to the diploid state being halved. So for example, you start with 1N, and then you form 2 cells, which is 1N, 1N, and then another 2 cells you divide to form 1N, 1N. So if you start with the 4 here, therefore it's going to be 2, 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 it's going to be 2. So it means that this is meiosis. Therefore, this is 1, this is 2. Therefore, the total number is 2. Please don't talk about 46 if they didn't say that this is the human cell. And then now they are saying original cell. So if it is original cell, therefore it's going to be 4. And then you see now they are saying, specifying it. Human cell in the sample diagram 2. Because diagram 2 is haploid, therefore it's going to be 20. It's going to be 23 chromosomes. So basically, that's how you're supposed to answer such questions. So you can answer these questions, at least in meiosis. What is A? A, in most cases, students confuse it with a nuclear membrane. This is not a nuclear membrane. You can't have a nuclear membrane when the cell is under a metaphase. Therefore, this is the cell membrane. This is the chromatid. This is the chromatid. This is the spindle fiber. And then this is the centriole. So this is metaphase, metaphase 1. While this one is anaphase, anaphase 1. Why? Because chromosomes are separating. What about this? This is the telophase, telophase 1. Why? Because they are chromosomes. They are chromosomes which are still double-stranded. Yes. Or at the beginning of prophase 2. Yes. So you see that now once you name that, you are able to answer this question. What is phase 1? We have seen it is metaphase 1. What is 3? We have seen it is telophase 1. Then you're saying the letter only of the part contains DNA, contains DNA. Give the letter of only part that contains DNA. It is B, because in this case they didn't mention any chromosome here. It's only B, which is chromatid, and then attaches the centromere of the chromosome, attaches the centromere chromosome, which is C, attaches is the centromere. Yes, and then they're saying that forms spindle fiber, which is the centrioles. Where is the centriole mentioned? Is D. You see, we are able to answer these questions. Name the organs in humans, male, where meiosis occur. In humans, male, it occurs in the testes. While in female, it occurs in ovary. In plants, it occurs in anthers, that is male, and then stigma, ovary in female. So here they produce sperms, they produce ovum or ova. Here they produce pollen grain, while here they produce ovules. Yes. So basically that's a simple illustration of meiosis occurring in plants and also in animals. Describe how meiosis leads to genetic variation. So now here you are talking about only processes which talk about genetic variation. Here you talk about crossing over. So you describe crossing over. Then you talk about the random arrangement of chromosomes during metaphase. Metaphase. Yes, so basically that's what you need to explain. It can lead to genetic variation. You can also mutation can also be part of meiosis, Why? Because if you talk about Down syndrome, it occurs during the process of what? Occurs in prosomers. But these two, they can help you to get those marks you are looking for. Here is an example of a karyotype. You have two individuals. Identify which karyotype. Here, when you're given a karyotype, look at, is there any abnormality? Which one is the male and which one is the female? So if you look at this, no abnormality, all of them, they are in pairs, but if you look at that, one is long and then one is short. Therefore, this is X, and then this is Y. So therefore, this is a male. And then both of them are the same size. Therefore, this is a female. So now from there, you can now go and then try to answer. What term is given the chromosome number 1 to 22? So we call them, because they can ask you to describe the structure of a karyotype, you call them orosome, the orosomes. And then 23, these are gonosomes. Why? These are sex chromosomes. Yes. State the gender of P. We have seen it. P. Where is P? Male, because of this. Give one observable reason, because the chromosome number, the chromosome number, the chromosome pair number 23. One chromosome is bigger than the other. Therefore, one is X, one is Y. Hence, the person is a male. Then they're saying give one observable. We have seen it. Each of the pairs shown is a homologous pair of chromosome. State the origin of each chromosome in the homologous pair of the chromosome. So here, where do they are two. Where are they coming from? These pairs, one is coming from the father, and then one is coming from the mother. Yeah, that's why there are two. That's why we say that when the sperm comes, is haploid, and then fuses the ovum, which is haploid. So one chromosome, another chromosome, when they combine, you form the pair. So it's the same thing. Sperm, ovum, sperm, ovum, sperm, ovum, sperm, ovum, like that. So one comes from the father, one comes from the mother, and then you form a diploid, a zygote. Then they are saying that the diagram below represents the distribution of chromosome 21 as it appears in the gamete. So it's the same thing, guys. Yeah, so you see that you always find almost the same diagrams, so same structures. So now draw a diagram to show the cell during anaphase 2 of meiosis. Now here, when I ask you to draw a diagram, you must write a title. That is the first thing, title. You cannot draw anything without a title. Use a sharp pencil. It must be neat. Yes. Someone is going to ask you, am I neat when I'm writing? Yeah, I'm saying you must be neat. I'm not a student. All right. I'm saying that. So when you draw, they're saying this is the prophase 2. Prophase 2, why? Because they are single. So what do? You draw the cell, and then they're saying during anaphase. So it means that before it's going to be like this, and then the short one down. So this is prophase. Just drawing a structure, but you have to show the variations when I'm drawing. So during anaphase, it means that one, and then one has a variation. So it's still moving to the poles, and then also then the short one is gonna be the short one, and then it's also moving. You see, but it also has the biggest variation. And then this other one is completely shaded. You see, you have to show that. And then this is spindle, this is chromatid moving, and then and then this is the cell membrane. Then write the title. Yeah, at least you are able to answer such question. Yes, so that when you come, we'll maybe give you a mark for title. Yes, a mark for drawing, correct drawing, and then maybe those are 2 marks. Now, maybe 3 marks for labeling. Yes, so that's why I'm saying labeled diagram. So MySys is also easy. If you look at most of the questions, you will see that we have answered those questions. Can you answer this? Yes. 10 seconds, 8 seconds, 7 seconds, 5 seconds, 3 seconds, 1 second. Okay, let's see. Then they're saying that this is what? Spindle fibers. What is this? Homologous chromosome. What is this? Centromere. Yes. So these are homologous chromosomes. And then this is the centriole. And then this is the cell membrane. So you can answer these questions. You see, nothing which is going to be new in your paper. Name the process diagram above. You see, it is metaphase. You see, you have to turn it so that these things are in the sides. Check. You see, they're in the sides. So that you see now clearly it is metaphase. Name the process, name the phase, represent a diagram. We have seen it. Then they are saying name part C. We have seen it also. Centromere. Then B represent homologous chromosomes. B represent what is homologous chromosome. Chromosomes which have the same structure and the same size, and then they carry genes. Genes of the same characteristics are always on the same locus. Yeah, so we call them homologous chromosome. Then they are not genetically identical, but genes of the same characteristic, they are in the same locus. Locus is a position of a gene. Is singular. Loci when they are many. Explain the appearance of chromosomes in diagram. Why are they shaded like this? Is due to crossing over, which took place in prophase 1, whereby the chromatid exchange the genetic material. That's why they are like that. They are saying that total number of DNA is the total amount of DNA is 12 viral units. Each daughter cell at the end of this cell division. How many DNA was in the parent cell? So they're saying that the total number of DNA is 12 units in each daughter cell. Remember, you start with the 2N. You start with what? 2N. Yes. So 2N, you form 2 cells, 1N, 1N, then you form another 2 cells, you form another 2 cells. So 1N, 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 1N. So if you started with the, if your daughter cells are having 12 here, so it means that then if you want to get the parent, is times 2. So which is 24. So the answer is going to be 24. 24 DNA binary units. Don't forget to put the units as they are being given to you. So many questions, you'll find them like this. This is the still a karyotype. Male or female is a female, while chromosome number 23 is having chromosomes which are of the same size and the shape. See? So describe the karyotype of person with Down syndrome. So a karyotype with Down syndrome will have an extra chromosome on chromosome number 23. Yes, so the pair chromosome, so there will be 3 instead of what? Instead of 2. So you see that these diagrams, these questions, you can answer them very well. Yes, you can answer them very, very well. All right, let's just do few questions about it. What is this? What is this? This is a centromere. What is B is a crossing over, point where crossing over takes place, therefore is chiasma. These 2 chromosomes under crossing over, we call it a bivalent. Bivalent or homologous chromosomes during crossing over. Then this, what is this? This is a chromatid, and then what is this? Is a chromosome. So these are sister chromosomes. So now you can answer the question. Describe the process that occurred at D. So it means that they're asking to describe the process of crossing over. I explained it before. This is where tricky things come from. They're saying the certain number species has 42 chromosomes. They're not saying it's human. So don't talk about 46. Yes, please don't talk about 46. They are saying 42. The muscle cell, in its muscle cell, remember muscle cells, those are autosomes. They're always 2n. Yes, give the chromosome number of each of the sperm cell. So it's going to be half because sperm cell is 1n. Therefore, the answer here is going to be 21. The skin is autosomes is 2n. Therefore, it's going to be 42. And then ovum of the female of the counterpart is the same thing. It's going to be 1n, which is 21. In most cases, the number of chromosomes are the one which are confusing students and they end up losing marks. So it's the same thing here. Crossing over took place. They can ask you to draw the diagram after crossing over. So members, students, please don't lose marks. Don't lose hope. You will make it. You will make it. I'm going to come back for genetics and inheritance. And then after that, I will go to human evolution. Thank you very much for watching. Don't forget to subscribe, like, and share. Don't also stop to comment so that I know where exactly I should put more effort and where to help so that you can get your distinction.
Please use the link below to get the notes and other videos.
Again, we have started a chapter of doing questions and answers, and then if you have any question, maybe you have a paper you want us to go through, you can send that paper to us, and then we can go through that paper as long as it is known, number one, SBA, because SBA we cannot go through SBA, but if it is something which is similar to it, then we can go through it, and then maybe give you ideas. SBA can only be done if it's a teacher who is asking for help. It's fine, you can send them to us. Any question paper you want us to go through, then we shall go through it. If you want the specific sections, then also we can also help you in that regard. Don't forget to subscribe to our YouTube channel, and then also to like and also to send a link to others so that you can also benefit. Don't forget that our channel is for science, geography, life science, and then even others will be coming, but we have started with grade 12, and then other grades will be coming. Only physical science and the life science is now with grade 10, but even other grades, other subjects are going to go to grade 11 and also grade 10. Grade 9 we are coming for you also. All right, the question is saying that various, in this case, please let's talk about some of the mistakes students do under objective. When we ask you to answer, they're asking you to answer A to D. So the answer is either A, B, C, or D. So when you are trying to answer such question, don't rewrite the question. Some students write the question. Number two, when the answer is A, for example, don't write A and then you write Darwin and then Lamarck. No, we only want the question is asking you A, B, C, or D. That's it. And then now once you write others, they don't even write A, they are writing the names of the, that's wrong, completely wrong. It means that you have not followed what? The instruction. Number two, questions regarding to objectives. You cannot leave it unattended. Why? Even you can guess. You never know that your guess can be correct and then you get a mark. You cannot leave a question which is an objective unattended. Even if they're saying that there is only 1 second remaining, just circle or just write A, you never know. You can get those 2 marks from that. Yeah, so you cannot leave an objective unattended. If I see a student, for example, on my case that has not attended an objective, I become furious. Why? Because that is a question. If you have not understood it, at least just write an objective, any letter which is between A and D. You never know, you can get it correct. All right, let's start. with the DNA. The structure of DNA molecule that was discovered by, the structure of DNA molecule was discovered by. So they have given you the name of scientists, the name of people who contributed to science based on our grade. So when you talk about DNA, remember there are 4 people who are involved. There are a lot of people who are involved in DNA, but based on our examination guideline, it's only 4 people: James Watson, Francis Crick, Maurice Wilkin, and then Rosalind Franklin. So you have to know that James Watson, they were together, and then Maurice Wilkin, and Rosalind Franklin, they were also together. You understand? James Watson and Francis Crick, they brought up the final DNA structure. And then Maurice Wilkin and Rosalind Franklin, yes, they're also together. And then they brought the initial stage of this DNA discovery. If you don't know, just look, check on our YouTube channel. The video for that is there. Or if you don't know it, you have failed to find it, just send us a message and then we will give you we'll give you the link for that. All right, so they're saying Darwin is not involved in DNA. Lamarck is not involved in DNA. These people are involved in evolution. So we're going to see these people where we're going to see them where in evolution. All right, so we see that Darwin and Lamarck, these ones are involved in evolution. So we don't see them in DNA. And what about Mendel? Mendel is involved in genetics. We see that Mendel is in genetics. Watson, yes, we see that Watson is there, but Watson is not with Mendel. So we can't give it a correct answer. And then what about Franklin and Wilkin? Yes, these people, yes, were involved in the DNA discovery. These 2 people, né? But this Wilkin, and then Franklin, they didn't discover the structure. They found out the initial stage of this discovery of the structure using the X-ray diffraction pictures, and then they took it to James Watson. So James Watson and Francis Crick are the one who tried to find out the correct answer. So the answer is going to be, we're going to find out that answer is D. Then we go to the structure of, we don't know which structure is it. This we might say that this is DNA, but may not be DNA. Let's find it, first analyze it properly. They said this is molecule 1, this is molecule 2. Yes, it means that these are different molecules. If you look at this, you find thymine. So you remember that thymine is only found in DNA, while this one is having uracil. And uracil is only found in RNA. So it means that structure A, molecule A is DNA, while molecule B is RNA. You see? So it means that now, yeah, this is what is the difference. So now, since you have got that information, now you can say an observable difference between molecule 1 and 2, an observable difference. Molecule 1 is double-stranded. Molecule 1 is DNA. We understand that molecule 1 is DNA, but it's not double-stranded here because they said observable. They want it, they want what you call observable. So if it is not observable, you can't see it here. So we can't classify it to be double-stranded. Then they are saying that molecule 1 is double-stranded, while molecule is single-stranded. Molecule 2 is single-stranded, we see it, but molecule 1 is not double-stranded, so it's not a difference there. Then they are saying molecule 1 contains deoxyribose sugar, and then molecule 2 contains the ribose sugar. It contains deoxyribose sugar, and then this one contains the ribose. But how do you identify it? You can't see it because they said observable. Then number 2, molecule 1 has thymine, while molecule 2 has uracil. Yes, you can see it by your eyes. Is this one is thymine, while this one is uracil. And then molecule 1 is longer, and molecule, there are two, the 2 molecules are equal. So the answer will become, we will take answer C as the most correct. Yes, as the most correct answer. Then we go to another question. The same diagram below shows the DNA profile. Yes, remember DNA profile is a technique. DNA profiling is a technique used to identify someone's DNA profile. It means that this person is unknown, but we have to compare it with a known one using a known DNA profile. The definition was removed from the examination guideline, but at least you have to know it before you go, because you can't deal with something which you don't know. So that's why we have to keep on giving you the definition so that you know exactly what is happening. Yes. So now they're saying that now you have, remember, you have to be with a ruler and then you draw so that you see which one is matching. So they're saying that the parent of Zintle and the Ayanda have 4 children, and then the 2 children are their biological offsprings, while the other 2 are not biological, meaning that they are adopted. So if they are 2 and 2, let's find out. The mother and the father, their DNA must match with the children. Okay, let's start. They're saying which of the 2 are adopted children of Zintle and Ayanda. So now we say draw. This one matches, but this one matches from the father. Then nothing, nothing. This one matches with the father. And then this one matches from the mother. This one matches from the father. This one matches from the mother. So this one is covered, meaning that this one is a biological child. Then we go to Gugu. Gugu, nothing, 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 nothing. So Gugu is not. So Gugu is not a biological child. Then you go to Bandile. Bandile, this nothing, nothing. This one matches from the Aya Zintle. Yes, and then this one also matches. Also, this is matches from Zintle. Again, this one matches from the father, and then this one matches from the father again, and then this one matches from the mother. So it means that Bandile is also a biological child. Then what about Li Tsue? Li Tsue. Li Tsue is nothing matching. It matches 1, matches 2, nothing matching here. This one matches, but not all of it. It means that this one is not a biological child. So it means that Gugu and this one, number 1, Gugu, Gugu, and this one are the adopted children of these 2 parents. So this one and this one, because they match with their parent, therefore we say that those are biological kids. So the answer is going to be the Bandi, Gugu, and so the answer is going to be B. Then they're saying that DNA molecule code for a protein containing 150 amino acids. Remember, we call it a protein. We call it a protein if it has 50 amino acids, amino acids and above. If it doesn't have 50 amino acids, then we don't call it a protein. We call it a polypeptide chain. We call it a what? A polypeptide chain. So that's how we call it. But if it is more than 50 amino acids, then we call it a what? A protein. So now they're saying the DNA molecule consists of 150 amino acids. Don't forget that each amino acid is being coded by 3 anticodons. Yes, 3 bases on the transfer RNA. So they are saying how many nucleotides, and each anticodon, each base on messenger, on transfer, on DNA, each base codes for a nucleotide. So remember, a nucleotide looks like this. Codes for a nucleotide. This is what call it a nucleotide. So now they're saying that how many nucleotides are likely to be present in the DNA molecule? Remember, they said that the DNA molecule code for a protein containing 150 amino acids. You understand? 150 amino acids, and each amino acid is coded by each nucleotide is coded by 1 anticodon, and then each anticodon, remember, each anticodon, if this is a transfer RNA carrying, it has 3 of them. If it is U, A, G, 3 of them, we call this one anticodon. If this is T, R, N, A, yes, and then this is amino acid. So if this is amino acid and this is transfer RNA, then it means that this is going to be the anticodons.
Yes. So if this is anticodon, the 3 bases on messenger, each base, you see, nitrogenous base, each base codes for and each base code for is like it forms what you call the nucleotide. So now it means that if I have 3 bases here, it means that I have 3 nucleotides. So it means that one amino acid is equal to 3 nucleotides. You understand? So now they're saying we have 150 amino acids. So how many nucleotides are there? So I'm going to say that I have 150 times 3. So it's going to give me 0 here, 0 here, 15 here, 15 here, and then I take 1, 3, which makes them 4. So it's going to be 450 nucleotides. So the answer is going to be B. That's how you can find it there. Sometimes we bring the nucleotides and you ask you how many amino acids are there. Sometimes we bring the bases, the codons, and then we ask you how many transferrin A are going to be there. So we can bring the question in a different angle. So now we have seen the answers of this. Now let's go to the terminologies. You cannot go to the paper without knowing the terminologies. The building block or the monomers of DNA. The building blocks. Now, next time I'm going to show you the booklet or what we have covered versus what it is here. So the building block of, or the building blocks or the monomers of nucleic acid. How do we call the monomers? The monomers. Remember, the monomers, it means that the bricks which make up the DNA. Nucleic acid is messenger RNA and is RNA and DNA. So now the answer here is going to be nucleotide. Nucleotide. Why? These are the nucleotides. Remember, nucleotide, we said it's this, which phosphate, sugar, and nitrogenous base. You are supposed to draw it nicely. Ne? A phosphate, we said, is a phosphate, a nitrogen, a sugar, depending on which, and then a nitrogenous base. So these are the building blocks. So we call this one a nucleotide. Then the gap in the nuclear membrane through which the messenger RNA leaves the nucleus. The gap, the hole. So it becomes the nuclear pore. Yes. So don't forget about that. That's where messenger RNA moves out of the nucleus and then it goes to the ribosome in the cytoplasm. Then the natural shape of DNA. Many students, they will say, double-stranded. It's wrong. It's not double-stranded. They are looking for the natural shape of DNA is double helix. We call it double helix. That's the answer we want. Nothing else. Double helical, it's not correct. We're supposed to say double helix. Is helical? No. We want double helix. Guys, learn how to give the correct answer without the examiner trying to figure out whether your answer is correct or wrong. Give the correct answer direct. What we ask you, what we give you is what you're supposed. Look at those slides I send you. I send you, or click on the link. You go then look for those slides on our website. Then you will find out the correct answer. Different color means marking point. Okay. Then they're saying that the sudden change in the nucleotide or sequence of DNA, change in the DNA composition, arrangement of nucleotides. We call it mutation. We call it mutation. But it depends. If it is genes, because they're talking about the sequence of the nucleotide, meaning that those are bases which are going to change. It's only a part of DNA which is going to change. And these parts of DNA, they code for, they determine what called a gene. So it means that it's only part of a sequence which changes, and it means that then it is a gene. So it's going to become what you call gene mutation. So change in the genetic composition of an organism, in science we call it mutation. But because it is happening in the gene, then we call it gene mutation. Then when we go to this structure, before you do anything, first identify which structure is that. Yes, first identify and then find out which structure is is that. Okay, now here, what we see here is we are seeing DNA, the structure of what? Of DNA. Why am I saying it's DNA? Because they are saying it here. You find out that what is it? Why is it saying DNA? But they said it here. Students tend not to read these instructions which are being given by the what? By the examiner. All right, identify the molecule. Identify the following. Molecule W. Look for molecule W. Where is molecule W? They're saying this. We say that this is a nucleotide. Why are we saying it's a nucleotide? Because we know that the circle means phosphate, and then this one means a pentose, or what you call its name is a ribose. But because it is DNA, then it's going to become deoxyribose. And then this is thymine, which is a nitrogenous base. So a phosphate, a sugar, and a nitrogenous base forms what you call a nucleotide. So W is a nucleotide. What about U? U, they are looking for the whole structure. What is it? Eh? And then they say that this is DNA replication. Definitely, the whole structure is DNA. Yeah, it's like they're asking you the answer which is already given. And then they're saying that parts of molecule WX, molecule labeled W, part of molecules W labeled X and Y. Where is X? X is a phosphate sugar. And then Y is a deoxyribose sugar. Don't say sugar, we won't give you. Don't say pentose, because we want to know. Pentose means 5-sided sugar, but we want to know which kind of pentose is that. So it's going to become deoxyribose sugar. And then bond Z. Bond Z is weak. You want to see the word weak. Weak hydrogen bonds. Yeah, if you write your hydrogen bonds, your answer will be having some questions, but we prefer you saying weak hydrogen bond. We call that one a KO, meaning that knockout. When you give the answer, no one is going to question about your answer. And then they're saying that where in the cell does this process takes place? Where does DNA replication takes place in the cell? Where does it actually? They have given you the answer because you know that DNA is only found in the nucleus, in the cell. If you are talking about DNA, this kind of DNA replication, leave alone the mitochondrial DNA, the chloroplastic DNA. Where is it found? It's found in the nucleus. It's asking you, where does the king found? It's found in the palace. Simple. So it's found in the nucleus. So this process occurs in the nucleus. Yeah, simple. And then they are saying that name the phase of the cell cycle during which replication takes place. Which part of the cell cycle? Where do we find this in the cell cycle? That's basically what name the phase of the cell cycle during which DNA replication takes place. Remember, what is the function of DNA? To double the chromosome number. When does the chromosome number doubles? It doubles before the cell divides. And which phase is it? It is interphase. So it basically, you can say that before the cell divides or during interphase. During interphase, then you'll be able to get the answer here. So interphase. Then explain the consequences if the type of bond Z is not able to break. What if these bonds don't break? What if? Definitely, you won't have two strands. Yes, it means that there is no unzipping. Yes, there will be no unzipping. So if there is no unzipping, it means that there will be no formation of new strand. And then it means that there will be no DNA replication. You see? So if it fails, it's like you have failed to open your trouser. What is going to happen? They're going to cut it. If you don't want to cut it, then definitely it means that you remain with your trouser. You can't remove it. So the trouser should unzip first so that now it can create a space for you to remove it. So if this fails to unzip, yeah, if it fails to unzip, it means that the new strand, this strand which is here, this strand will not form. And also this strand will not form because we will not have a mirror on it. So therefore, we say that DNA replication will not take place because the two strand will not form. There will be no unzipping. Yes, and then it means that the new strands will not form. So basically, that's what you can say. And then lastly, on the DNA molecule, if you bring such a diagram, this is a diagram which shows the protein synthesis. Protein synthesis. So basically, they're saying the diagram below shows the protein synthesis. They have mentioned it. Whatever happens in the nucleus, that is transcription. Yes, transcription. And then what happens in this is the nuclear membrane. What happens in the outside in the nucleus, then is the cytoplasm, then that is translation. So you have to know this. You have to know that. Then now they're saying that what is X? X, you know, that's DNA. You have to label them maybe before you start. And then what is this? This is a messenger RNA has been formed. And then what is this molecule which looks like that? It's a ribosome. And then basically that's it. And then now you can start answering the question. Identify molecule X. We have seen it. We said that this is what? This is DNA. Ne? We have seen that that's DNA. Organelle Y, organelle Y. What do we call it organelle? Because it's double membrane. Whatever structure is double membrane, we call them organelles. So now, organelle Y is ribosome. The body, zome means the body. Ne? Ribo is coming from the word ribose, ribose. It is a ribose sugar, so it means that it's a body made up of ribose. That's why we say that the RNA forms the ribosome. Why? Because ribosomal RNA, it forms the ribosome. That's why we call it is a body which is made up of ribose sugar. And remember, this is a ribose. All right. Nitrogenous base 1. Nitrogenous base 1. Where is nitrogenous base 1? Nitrogenous base 1 is here. If this is cytosine, then definitely this is going to be guanine. Then they're saying describe the role of DNA during transcription. Then you define what's transcription, formation of messenger. So DNA forms the messenger, and then this messenger code for specific code for the protein. It carries, the messenger carries the information from DNA for protein synthesis. So basically, DNA forms messenger which carries information for protein synthesis. Yes. So if we don't have DNA, we don't have protein synthesis. The table below shows the amino acids that correspond with the different DNA molecules. They are saying adenine, arginine, methionine, glycine, and then arginine. So they are saying they are giving you. So before you start anything, first identify what is here and what is being given. So write down the correct sequence of amino acid. Now you see, they're looking for amino acid. Sometimes this is messenger, sometimes this is transfer. And sometimes don't say messenger transfer. No, we say codons and anticodons. You have to know that when you say code, we are talking about, you see, we are talking about DNA. We are talking about DNA. When you say codon, we are talking about messenger RNA. When you say anticodon, we are talking about transfer RNA. You have to know this because we also going to, sometimes we use this language to bring about that. So now write down the correct sequence of amino acid on the structure S. Then we go back to structure S. Where is structure S? Where is structure S? I don't see structure S here. Structure S. Where is structure S? Yeah, structure S is this. So now you have, it means that they are saying DNA. Because here in the table, they are showing you, here in the table, they are showing you code. It means that what we are going to do here, we are going to change this into DNA so that now we can read it here. We can read it from the table. So I have A goes with what? A goes with T, and then U goes with the A, and then G goes with the C, C goes with the G, and then again C with the G, and then A goes with the T, and then A goes with the T, and then G goes with the C, and then A goes with T. So now I'm gonna say this is 1, this is 2, and then this is 3. So now I come here, and then I find out which one is 1, which one is 2, which one is 3. ATC. I come here, I look for ATC. So the first one, this one is going to be number 1, and then I have GGT is gonna be glycine is gonna be number 2, and then I have TCT is gonna be number 3. Then you write them in order. Then you get your marks. Guys, I know that this part always disturb student. If you really need more question, or just comment down in the comment. Just comment down that you need more help on this. Then we shall help you, or we shall do more classes concerning about that. Then now we have what we call if the 9th base on molecule S was changed to C. If the 9th base on molecule S was changed to C. Yes. And then explain the consequences, what will form. They're saying 9. So if I have, this is 1, 2, 3, 4, 5, 6, 7, 8, 9. So if this was changed to C, so it means that what I'm going to form T, C, and then this A was changed to C. So it means that A was changed to C. It means that it's gonna have G. So I look for TCG. TCG is arginine. But the third base was this. It is arginine. Also, this is also coded for arginine. Since it codes for the same amino acid, then it means that there will be no change in the protein, and then this will not change the functioning of the organism. But if this code for another amino acid, then it means that you're going to form a different protein, and even the functioning of the organism is also going to change. But because the one which is being replaced is the same as the one which was original, 3 is the same as the new one, then it means that there will be no change in the protein and they will remain the same. So basically, that's what you are supposed to know. It's like asking you, calling you Karabo, and then the same time you are, for example, you are Kabero, for example. I'm just giving an example. So if I call you Karabo, you say yes, sir. If I call you Kabero, you call yes, sir. But if you are something else, maybe you are Tapero, and then I call Tapero, and then you are not Tapero, then it means that you will not respond. So it means that the functioning will change. If it codes for the same amino acid, however much the base has changed, then we say that the DNA is not going to, the protein is not going to change, and the functioning is not going to change. But if it codes for a different one, then it means that the functioning is going to change. Guys, I love you so much. I'll see you next time. We're going to look at other questions concerning about meiosis. If you have any question, drop it, or WhatsApp us. Then when we go to the general discussion, then we can discuss about it. Thank you very much. I'll see you next